Hi everyone, am I audible? Hello? Hello? Am I audible? Hi everyone, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today we are going to finish molecular basis of inheritance. So welcome everyone, happy morning, happy Sunday. How are you? Good morning, good morning, good morning. So first of all, tell me, am I perfectly audible and visible to you all? Am I perfectly audible and visible to you all? Hello. Sorry to disturb you on Sunday morning, but you know that we really need to finish molecular basis of inheritance, right? And I hope now you guys are clear about the biology syllabus, about your NEET 2024 syllabus, right? Me, Wazeem sir, Shreyas sir, we have shared many videos with you all so that you guys can get the clarity that what exactly you need to study for your NEET 2024. Okay. So here you people can see, right? See, this is the video that, this is a session that I took yesterday. So here there is a complete strategy. So bache, if you have checked it, good. Otherwise, see, otherwise what you have to do, check out this video. In this video, right, I have mentioned that which topics are added in your syllabus and which topics are deleted. And see here also, we are discussing what? Molecular basis of inheritance, right? Molecular basis of inheritance. So you should check this. Fine, you should check this. So have a look here. See this. We have completed this part. So today we are going to start from genetic code, right? Translation, gene expression. This is what we need to discuss. So as per NMC, right, in your molecular basis of inheritance topic, they have added one extra point, that is protein biosynthesis. Okay, that's what they have added extra. And don't worry, we will even complete that. Done? So do check out these videos, and please, now you have a proper plan. You have that 75 days challenge, and I told you what you have to do, how you need to proceed. So you have 180 plus days, or let's keep it 180 only, and you have 32 chapters of biology. And trust me, even if you will give 5 days to each chapter, still you can complete your syllabus and you will get time for the revision as well. Okay, revision as well. New NCERT or old NCERT. See, bache, there are certain topics which are not even there in the new NCERT. So I think as of now, for this particular year, you need to keep both new as well as old. Okay, anything else? Anything else? And in this video I have provided the details, so do check out this. Okay, so now let's start the class, and let's start with the genetic code. So before that, let's revise quickly what we have covered so far. So when you talk about this chapter, that is molecular basis of inheritance, right? In this chapter, that is your molecular basis of inheritance, bache, you need to first start with the DNA. Isn't it? You need to start with what? You need to start with the DNA first. So DNA, and then comes the detailed structure of DNA, its discovery, everything you need to cover, right? You should know about the RNA here, then DNA replication, and before DNA replication, even the DNA packaging, okay, and then the semi-conservative mode or semi-conservative model of DNA replication. I'm using the short forms as of now, right? So we are done with that too, and the transcription done. Okay, even transcription done, even the types of RNA done. That's what we have covered so far. Okay, so today we need to start from the genetic code. Understand the genetic code first, and then the translation, operon, and human genome project, DNA fingerprinting, and the chapter will be over. And the new topic that they have specifically mentioned in your new syllabus, that is protein biosynthesis, I will keep a separate session and then we will discuss it. Is that clear? Right? And yes, for the next week, bache, this is what we are going to do, right? That's what we need to follow now. Every week I will post one video and I'll tell you this is what we are going to cover in this week, and you have to prepare accordingly. Okay, with marks, I will tell you that if I'm going to pick 4 chapters in a week, right, I'll tell you the marks, and that's what we will cover. Okay, and this time I'll make sure right? Whatever I'm going to commit you, I will fulfill that. Fine. Like sometimes happen, some emergency is there, and because of that we have to end the session, or we have to postpone the session. So I will keep all the factors in my mind, and then I will provide you the videos, and we will decide that this is what we are going to complete in a week. But what I need your support. I need your consistency. I know it's difficult that still we have 6 months. Why do we need to spend so much time, right? Why do we need to watch such long lectures, right? We can do it later also. Teachers are going to take the one shot. We can do it later. But that's not the right way actually. Still you have 6 months. If you will read things in detail, you can master your topics, okay? And now, because you know in biology, your chapters are less, right? So now the competition will be more. I'm not saying the paper will be difficult. No, it will be moderate as per me. But let's see, right? I cannot predict it honestly, right? But what can we do? We can just prepare for that paper. So yes, the competition will be more now. Okay, the competition will be more now. Clear? So you have to prepare accordingly. So now let's start, and let's start with the genetic code. So you tell me, what do you know about the genetic code? What do you know about the genetic code? It's something very important. And you know, the term was given by George Gamow. It was given by George Gamow, this term, when you talk about the genetic code. And if you talk about the scientists, those who discovered it, they are Nirenberg, Mathai, right? So they discovered it, Nirenberg, Mathai, Dr. Hargobind Khorana. Okay, so they discovered this genetic code. Are you getting it? Yes, bache. Alpha, I have already answered your question. So please do not spam here. Please do not spam here, right? And there is no deduction from morphology and anatomy part, and even in morphology they have added some extra families, so you have to prepare accordingly. So just have a look, guys, just have a look. So as I'm saying, the term was given by George Gamow, and when it comes to the discovery of the genetic code, so it was discovered by, the name of the scientist is important, Nirenberg, Mathai, and Dr. Hargobind Khorana. Okay, Dr. Hargobind Khorana. Obviously, he's an Indian scientist. He was an Indian scientist. Clear, bache? Clear, bache? So what exactly this genetic code is going to explain? You know about the central dogma, isn't it? You know about the flow of information. You know that it is the DNA that will form mRNA, and that mRNA will form the protein, isn't it? DNA is going to form the mRNA, and it is the mRNA that will form the protein. So DNA to mRNA is your transcription, and mRNA to protein is your translation, right? So now look at it very carefully, bache. mRNA is actually like a primary transcript. It is just like we are copying something from DNA, right? It is just like you are copying the information from the DNA. That's what your mRNA is, isn't it? Now, if you talk about the DNA, it is also made up of polynucleotides. Am I right? Your DNA is also made up of polynucleotides. When you talk about the mRNA, it is also made up of polynucleotides, isn't it? Even it is made up of polynucleotides, isn't it? DNA is also a chain of polynucleotides. mRNA is also a chain of polynucleotides. But when you look at these proteins, right, they are what? They are also the polymers. Protein is also a polymer, but it is a polymer of amino acids, isn't it? Proteins are made up of what? Proteins are made up of amino acids. So how is it possible, Priya, how is it possible, right, that polynucleotides, right, a long chain of polynucleotides, or you can say that a chain of that nitrogenous bases, how is it possible that they are making the amino acid? Right, this is the flow of information, right, Vengi? This is the flow of information. So from polynucleotide, how is it possible to get the amino acid? Actually, that relationship is explained by this genetic code. Okay, Suhashini, this relationship, okay, between the sequence of amino acid in a polypeptide chain and in between these polynucleotide, right, and in between this polynucleotide sequence, actually it is basically your genetic code. Is that clear? It is basically your genetic code, and that's what we need to understand here. Done, bache? So this mRNA, it is having the bases, and that bases are going to code for the amino acids, and that amino acids are going to form our protein. Fine, that amino acids are going to form our protein. So ultimately in our NCERT, we have a table, right? See, I have added one. So this is what we need to understand first. Done, bache? This is what we need to understand first, that what is this table and how, how can we explain this part. Done. So yes, one question can come from this portion, and today I will tell you how to make this table in an easy way. Done, bache? Right? So this is what we are going to discuss. Okay. So now when we are talking about the mRNA, as I said, it is having the basis. Let's say it is having a something like this, right? Randomly I'm taking a sequence. Okay, let's say this is what your mRNA is having. So actually, bache, what is happening, right? On this mRNA, you know that you have these nitrogenous bases. So we take the sequence of 3 bases. Do you know that? We take the sequence of 3 bases. How many bases? 3 bases. How many bases? 3 bases. So 3 bases are making one codon, and that one codon is giving us one amino acid. That's the story actually. What is happening here? You are having the codons here, right? You are having the codons here, right? It is actually a triplet base codon. Why triplet? Because this codon contains 3 bases, and that 3 bases are going to form what? That 3 bases are going to form one amino acid. That 3 bases are going to do what? They are going to form one amino acid. So that's what you need to keep in your mind. Okay. Okay. So that's what we study here in the genetic code. So we need to discuss the properties of the genetic code as well, and that properties of genetic code, that topic is important for boards as well. It is important for the NEET as well. Okay. It is important for the boards as well. It is important for the NEET as well. Clear, bache? Clear, bache? So that's what they discovered. But the question is how? Okay. So I'll ask you one question. See, when you talk about the bases, how many bases do we have? How many bases do we have? When we are talking about the bases, how many bases do we have? How many bases are there on your RNA? Quick, answer it. Of course, 4. Adenine is there, guanine is there, cytosine is there, right? And uracil is there. Okay, but when you talk about the amino acids, the protein forming amino acids, bache, protein forming amino acids, they are 20. They are 20. Remember, alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, right? Serine, threonine, tryptophan, tyrosine, and your valine is there. So basically, how many amino acids you have? 20. Now we include 2 extra as well. Okay, we include the pyrrolysine, we include the selenocysteine as well, but we have to go as per NCERT. So 20 amino acids are there. But now we have 20 protein forming amino acids. They are 20 protein forming amino acids. We have near about 100 to 200 amino acids. But when it comes to the protein forming amino acids, they are 20 in number, Subhashini. Right? They are 20 in number, but the bases are 4. So we have to take the combinations. It's very simple. Right? It's very simple that we have to take the combinations. See, if we will take single base will give us single amino acid. So you will get only 4 amino acids, which is not possible because you need 20. And even if you are taking the sequence of 2, okay, even if you are taking the sequence of 2, right? If you are considering that your genetic code is doublet, right? Even if you are considering that your genetic code, it is doublet. Okay, doublet means 2 bases are there. So this position can be filled by 4 different bases. This position can be filled by 4 different bases. So you are getting 16 combinations. But still, again, it is not fulfilling our requirement. Right? It is not fulfilling our requirement. 16 combinations means 16 amino acids. Now, if let's say your genetic code is triplet, right? If it is triplet, means 3 bases are there. So, see, that 3 positions can be filled by 4 different bases. I hope you know little about the permutation combination part. Okay, so at this point, out of that 4 bases, any base can come. Here it can be adenine, guanine, cytosine, uracil, anything. Here also, here also. So 4 into 4 into 4, it is going to be 64. So yes, 64 codons can solve our problem, isn't it? Isn't it? This can solve our problem, isn't it? So if we consider that our genetic code is a triplet, that is one codon is having 3 bases, then how many codons you are going to get? You are going to get 64 codons. It can solve our problem, isn't it? It can solve our problem. See, we have 20 amino acids. 64 minus 20, it will be 44. So still you have 44 codons left, isn't it? Still you have 44 codons left. So 64 codons are sufficient to code, right? They are sufficient to code 20 amino acids. Are you getting it? They are sufficient to code 20 amino acids. Am I right? Yes, bache. Gokul. Am I right? Okay. So out of these 64 codons, fine. Out of these 64 codons. So codon means it is a triplet. Triplet base codon means it is having 3 bases. So out of these 64 codons, what's the point? See, 3 codons are stop codons. 3 codons are what? They are the stop codons. So codons are nothing. These are just the sequences of the nitrogenous bases present on mRNA. So what is the meaning of stop codon? It is something which will stop your translation. It is something which will terminate, which will stop that protein synthesis, which will terminate that protein synthesis. Okay. So whenever, because when it comes to the translation, you are going to talk about the ribosome. You are going to talk about the mRNA and the tRNA. So when that ribosome, when it moves over that mRNA, so when that ribosome, when it finds this stop codon, any of the stop codon, they are going to stop the translation. It's like a signal. So out of these 64 codons, 3 codons are stop codons. So 64 minus 3, 61 codons. So if I take the reference of these 61 codons, directly you know that you have 20 amino acid. So minus 20, what it will be? 41. 41 codons are left. So can I say that these 41 codons are also coding for the same amino acids? These 41 codons are also coding for the same amino acids. Can I say so? Can I say so? Of course I can. Of course I can. Right? So from this information, can I say that for coding the amino acid, for coding a particular amino acid, there is a possibility that we can have more than one codon? Can I say so, Subhashini? Let's say if I'm talking about a particular amino acid, let's say if it is phenylalanine. Remember phenylalanine? We convert phenylalanine into tyrosine to form melanin. I hope you remember. Right? So let's say we have one amino acid which is known as phenylalanine. So phenylalanine can be coded, we can code this phenylalanine with the help of different codons. Okay. Okay. So from this information, this is what we can explain. Like you can say that it can, I'm just giving you 2 examples only. Right? I'm just giving you these 2 examples only. Is that clear? Is that clear? Yes? Tell me, is that clear? Okay. Okay. So if we have, say out of these 61 codons, 20 so we can directly say they are forming the amino acid. Rest 41, they are also coding for the same amino acid. So that's why we are saying that one amino acid can get coded by more than one codon. Right? More than one codon. Done? So that's what you need to keep in your mind. So now when it comes to the stop codon, they are also known as termination codons. They are also known as nonsense codons. Okay, nonsense codons. So they are basically this. So it is also important. Done? These are that 3 stop codons. Okay, these are that 3 stop codons. So how to remember this? See, this is my trick. U. Right? It's very simple to remember that stop codon, they start from U. So U. So I remember it like this. Ag, aga. Ag, aga. That's my way to remember. Okay, it's Ag Aga. Ag Aga. That's how I remember. Done, bache? So you have these 3 stop codons. Clear, bache? You have these 3 stop codons. So presence of 3 stop codons indicates that now the translation, it will stop. Done, bache? It will stop. Done. Done. So there are another names as well. Like this one is your occur. This one is your, okay, umber. And this one is your opal. Is it right? Right. So umber, occur, occur, umber, opal. Done, bache? Occur, umber, opal. So that's what you need to remember. Done. There are another names as well. That's important. Fine, that's important. Now let's talk about the discovery part, and that we will do from the NCERT, and then I will tell you about the table as well. I'll tell you about the contribution of the scientists here, and yes, in NEET examination you used to get such type of questions. Fine, right? So here you guys can see that during replication and transcription, a nucleic acid was copied to form another nucleic acid. Of course, understood. DNA was forming mRNA. One nucleic acid was forming another nucleic acid. Right, bache? So hence these processes are easy to conceptualize. Okay, bache? Why? On the basis of complementarity. So bache, if I will be the examiner, I can ask the question from this point. Right? That how can you explain the formation of one nucleic acid from another? Simple, because of complementarity. It is the complementary bases which are helping in this synthesis. Right? It's a very simple question, simple answer. But when it comes to the translation, what is translation? Formation of proteins from the mRNA. Right? So here you are transferring that genetic information from polynucleotide chain to a polymer of amino acid. So this is what we explain with the help of this genetic code, Subhashini. Clear, bache? So see, in the classes we are also covering NCERT as well, bache. Right? I'm no doubt, I'm not going this, I'm not giving a different name to this series. That see, we are, completing NCERT, it is NCERT highlight. No, we are not doing it like this. But still we are covering. Okay, still we are covering. So please be attentive in the classes, because after finishing our syllabus once, when we will start, when we will start the revision, then I will only keep NCERT there. Okay, I will only keep NCERT there. So kindly focus. So see, in the polymer of nucleotide and amino acid, there is no complementarity. Right? Right. But how is it possible? How is it possible? So here you guys can see there existed ample evidences though to support the notion that change in nucleic acid, right? Scientists, they have studied it, that if there is any change in nucleic acid, it is changing the amino acids in the protein. If there is any change in nucleic acid, it is changing the amino acid in the protein. Okay, so this led to the proposition of the genetic code, bache. Fine. Fine. And now, if determining the biochemical nature of genetic material and structure of DNA was very exciting, this proposition and deciphering of genetic code were most challenging. It was not that easy, right? Scientists conducted many experiments, Gopika, right? They have conducted many experiments. Are you getting it? So in a very true sense, it required involvement of scientists from several disciplines like physicists, organic chemists, biochemists. So all of them, they played the role here. So as I said, the term genetic code was given by George Gamow. Very important. He was a physicist. Are you getting it? He was a physicist. So that's important, right? That's important. Right? So George Gamow argued that there are only 4 bases, and if they have to code for 20 amino acid, the code should constitute a combination of bases. So from this line also they can directly pick up the question that who argued that if 4 bases need to form the 20 amino acid, they should be in combination. So the answer is yes, bache. The answer is yes, everyone. Type the answer in the chat section quick. Answer is George Gamow. Right, the answer is George Gamow. Clear, bache? Clear, bache? So after applying that permutation combination, we got to know about the 64 codons. Clear? Clear, clear, clear. But finally, the proof that codon was triplet was a more daunting task, and chemical methods to prove this was developed by Hargobind Khurana. Right, that's important, bache. Their discoveries are important. Important MCQ it is. Fine, it is important. So see, Hargobind Khurana developed a chemical method, right? See, So the chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with a defined combination of bases. So Hargobind Khurana, he took homopolymers and copolymers, right? What is the meaning of this line? The homopolymers and copolymers. Do you know that? What is the meaning of this part? That he took the homopolymers and copolymer. Homo means same. Like let's say if I'm taking all the same bases. So again, which amino acid will I get? Phenylalanine, phenylalanine, phenylalanine. Now let's say, I'm just giving you example, bache, random example. Let's say it's this, copolymers. Let's say it's this. So CUC is coding for, see, the CUC, it is coding for leucine. That's what I'm saying now, CUC, UC, CU, something like this. So see, this CUC is coding for your leucine and UCU is coding for phenylalanine. Then again, leucine, phenylalanine, right? So they observed such results. Like when they were taking when they were keeping all the bases same, they were getting same type of amino acid. But when they started using the copolymers, let's say they took two nitrogenous bases and repeated it like Cu. But you know that, right? That when it comes to the formation of amino acid, 3 bases will be considered. So obviously you will get leucine, then you will get phenylalanine. Leucine, phenylalanine, like this. So they conducted different experiments. Okay. Okay. So you have to remember that it was the experiment of Hargobind Khurana. Now the next part, Marshall-Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. So what Marshall-Nirenberg, what he did, bache, he formed a cell-free system so that they can synthesize that proteins, so that they can understand that which code is coding for which amino acid. Sorry. See, now it's just like a puzzle. You have the bases, you have the combination, but you don't know that which combination is going to form which amino acid, right? So they experimented different things. Okay, then they concluded ki, okay, fine, this codon is forming this amino acid, this codon is forming that amino acid, like this. So here the Nirenberg cell-free system helped a lot. Done, bache. And then comes your Severo-Ochoa enzyme, again important MCQ. Right, genetic code itself is very important, right? For sure you will get question from genetic code, you will get question from Human Genome Project, for sure, right? Definitely they will ask you the questions from these 2 chapters. So Severo-Ochoa enzyme, the name of the enzyme is polynucleotide phosphorylase. The name of the enzyme is polynucleotide phosphorylase. Are you getting it? So it was also helpful in polymerizing the RNA with the defined sequences in a template independent manner. So this enzyme of Severo-Ochoa, the enzyme is your polynucleotide phosphorylase. It was helpful in forming RNA, bache, in template independent manner. As of now, you know that we need the DNA, and from that DNA, the information will be copied and you will get the RNA. That's how the story is. That's what the story is. But here this enzyme, that is polynucleotide phosphorylase, important question it is, bache, right? That's why I'm teaching it from NCERT. Okay, so this enzyme helped to synthesize the DNA in a template independent manner, right? So now you do not need the DNA and then you have to form the RNA. No, directly with the help of this enzyme you can form the RNA. Clear, Nandini? You can form the RNA. Done, bache? And finally, they prepared this checkerboard. Okay, so I will tell you about this checkerboard. So how many of you find this checkerboard difficult? That, ma'am, we don't even understand what this checkerboard is. Tell me. Tell me. How many of you find it difficult? And trust me, it's easy. Trust me, it's easy. I'll give you one example here. That's your mRNA. The direction of synthesis is 5' to 3'. The direction of synthesis is 5' to 3', yes or no? So let's say here you have certain bases. But see, there is a sequence on mRNA, right? When we need to form the amino acid, we have to read the sequence in one particular direction. It's not like that you can start it from 3' or 5'. No, always you have to start it from 5' towards the 3'. From 5' towards the 3'. So this should be your reading frame. Okay, this should be your reading frame. So you will pick up your bases, first 3 codons you are going to pick, right? And then there will be then you have to pick the next 3 codons. It is the unique property of this genetic code. Do you know that genetic code is universal? Even if you are talking about any unicellular organism, right? You will see that same type of codon is forming same type of amino acid. Do you know that? Same type of codon is forming same type of amino acid. That's what you are going to observe there. Okay, that's what you are going to observe there. That's very important. Okay, that's very important. So initially 3 bases, 3 bases, 3 bases, like this you have to pick. So first point that you have to remember is that your genetic code is universal. Even if you are talking about the unicellular organism, the same codon will form same amino acid, and even if you will see that in your body, you will get the same results. There are only few exceptions, and that exceptions are your homework. You will tell me about that exceptions. First point. Now come to the second point. The second point is saying that there is no requirement of any punctuation in this genetic code. There is no need to put comma. There is no need to put full stop here. Okay, I will read it from NCERT as well. Right now, when you are taking the codons, you will take first 3 bases, then next 3 bases, then next 3 bases. Remember, you cannot do it like this. Like let's say here the sequence is this. Firstly, you are picking AUG. Then you are picking UGC, then you are picking GCU. This is incorrect. This is incorrect. You cannot read it like this. You have to pick first 3 bases, then next 3 bases, then next 3 bases, then next 3 bases, then next 3 bases. It should be like this. It should be like this. Clear, bache? Clear, bache? Are you getting it? So here, what I was telling you, the reading frame. So the reading frame is 5' to 3'. This is your reading frame, right? This is your reading frame, 5' to 3'. So how many bases do you need to consider? 3 bases, right? So this will be your first base, second base, and the third base. Okay, and the third base. Clear, bache? So now when you look at this checkerboard, this side of the checkerboard is showing your first base. Do you know that? That is actually the 5' end. This side of the checkerboard is having your second base. Okay, and this side of the checkerboard is having your third base, bache. So it means it is the 3' end. It is the 3' end. Clear? It is the 3' end. So now another trick is you can arrest gangster. That's what you need to remember. You can arrest gangster. You can make the better one as well, but this is my trick. Okay? So the trick is you can arrest gangster, right? That's the trick. So you can means U, then C, then A, then G. So you have to write it here in this sequence only. Okay, like U, this is for C, this is for A, this is for G. Here also U, C, A, G. But here, how are you going to write it? U, C, A, G. U, C, A, G. U, C, A, G, U, C, A, G. Trust me, it's very simple to make this table. It's very simple to make this table. So the first base is U here. The second base is also U here. Am I right? Am I right? Let's talk about this part. Let's talk about this part. The first base is A here. The second base is C here. So you have to write it like this. Okay? Okay? So now you answer me. Which one is the first base here and which one is the second base here? Tell me quickly. Which one is the first base here? Which one is the second base here? Quickly, Nandini, Priyanka, Trisha. Quick. Very good. Very good. The first base is C, the second is A. Okay. So what about this one? Tell me about the first and the second base. Tell me about the first and the second base. Tell me about the first and the second base. Gopika bache, you should start first, and if you will work hard, definitely you will get good result. Tell me. So exactly here it will be G and G, right? So see, when it is CA, right? So now when you are talking about the third base, it will be same. You can arrest gangster. Here also you can arrest gangster, means UCAG. Here also UCAG. Okay. Here also it's GG na, then UCAG. So you can keep first two bases like this. So now see this one. First base is A, second base is U. First base is A, second base is U. And then you can arrest gangster, UCH. That's how you have to make the table. It's very simple. It's very simple. Now come to this part. The first base is A, the second is G. First base is A, second is G. Right? And the third base will be in the same sequence. UCAG. Can you see that? UCAG. It's actually very simple. Okay, now come to this part. First base is AA, then UCAG, and you know that. Okay, so here it is. So now tell me, if there is any doubt, do let me know, bache. Tell me if there is any doubt. It's easy, isn't it? It's very easy. So that's how you can make this checkerboard. See, it's very simple. It's actually very simple. Now I will tell you about the amino acids. So no doubt they can ask you the question, they can give you a particular codon and they can ask you that it is forming. Yeah, they can ask you that this particular codon is forming which amino acid, right? But okay, second column is wrong. Which one? Tell me. Tell me. Okay, got it. That here, ma'am, it should be U and then C. Done. Okay, thanks, Risha. Done, bache. Done. Fine. So that's how you can make it. Now, wait. So see, it's as simple. It is very simple. So now see, here my trick is Sir, Pro, Thar, Allah. Right? Like from their initials, na, I memorized it. Basically, I crammed this part. Right? I crammed this part. So it's Sir, Pro, Thar, Allah. Right? It's Sir, Pro, Thar, Allah. Means serine, all the four, see, these all four codons, four different codons are coding for same amino acid, serine. This one is proline, of course. This one is threonine, of course. And this one is alanine. Am I right? Isn't it easy? Don't you think it's easy? Yes, It's serprother ala means serine, proline, threonine, alanine. Right? From their initials, I memorized it. Okay, from their initial, I memorized it. Done. So that's how you can remember. So yes, in the paper, there is a possibility they'll give you the codon and they'll ask you that it is coding for which amino acid. But yeah, again, the chances are less. And obviously, it takes a lot of energy to remember this checkerboard as well. It's easy to make this checkerboard, but you don't remember which codon is making which amino acid. So what you can do is you can just make your own tricks. Like my trick here is serprother ala. So serine, proline, threonine, alanine. So if they will ask any codon, so first of all, I'll make the checkerboard and I'll immediately check that whether it is here or not. Right? And there is no need that you have to make that big checkerboard. If you understand that how to make it, then it will be easy for you. Right? It will be easy for you. Like here, it is for the phenylalanine. Right? And see, I told you now, about see, I told you about the stop codons as well. These two are the stop codon. There is one more. Can you tell me where it is? There is one more stop codon. See, this is also a stop codon. So you should know about the stop codons, right? You should know about the stop codons. You should know about the start codons. Okay, so at least remember these codons. Okay, at least remember these codons. Like these three are the stop codons. Now AUG. It's your start codon. This codon is having it is having dual purpose. It is coding for the methionine. It is coding for the methionine, bache, and it is also the start codon. So whenever we talk about the protein synthesis, always it will start from your AUG. AUG is the start codon, right? AUG is what? AUG is the start codon. Okay, and here, bache, if you will see this part, UGG, you know, it is coding for tryptophan. TRP means tryptophan. TRP means tryptophan. Okay, so see, like methionine, it is only coded by AUG. Tryptophan, it is only coded by UGG. So that's what you can remember, that there are certain amino acids which are coded by only one codon, right? There are certain amino acids which are coded by more than one codons, right? So such points, right? You need to focus on such points, right? So AUG is the start codon, right? So it is coding for methionine. It is, yes, also known as initiator codon. These three are stop codon, nonsense codon. UGG, it is coding for tryptophan. So tryptophan here, this is what you can remember. Tryptophan is also coded by only one amino acid, only one codon. Clear, bache? Clear, bache? So come to this part, your NCERT. See, phenylalanine, leucine. And see, this is also leucine. So leucine is coded by 6 different codons. Isn't it? Isn't it? Yes or no? Isn't it? Right? So it is the leucine amino acid which is having maximum codons. It is having maximum codons. No, bache, Shubhankar, it is not the lecture 1. I think it is the lecture 4th or 5th. Done, bache? So that's what you need to remember. Important it is. Okay, important it is. See? So remember this valine, leucine, like this. So serine, proline, threonine, alanine. I told you. So these two are for tyrosine, then stop codon. Histidine, right? Glutamine, asparagine, lysine. Right? This is leucine, this is lysine. Don't get confused. So aspartic acid, glutamic acid. Okay. So cysteine, the stop codon, the tryptophan, and then. So obviously, right? Even it is easy. So only methionine and tryptophan, only methionine and tryptophan are coded by one codon. Can you see that? Only they are coded by one codon. So this is kind of unique points that you can notice, right? That you can note down. Fine, bache. So leucine is having maximum codons, that is 6 different codons, right? Tryptophan and methionine, they have just one codon. For methionine, it is AUG. For tryptophan, it is UGG. Okay, for tryptophan, it is UGG. Fine. So this part, for this part, I have already given you the trick. It's Sir Protharpala. Right? Rest you can make your own tricks and you can revise. Done, bache? Okay. Done? Is it clear? So this part clear? This part clear? Sure? Sure? Fine. So try to make your own tricks here. So that's about, so see, this is the first base, second base, third base. Okay, that's all. So any other doubt? Yes. Do you have any other doubt? So now one more point is, bache, no doubt it is your AUG. Okay, it is your AUG, which is the start codon and the initiator codon or the initiator codon. But sometimes even GUG can act like a start codon. Do you know that? And GUG codes for what? GUG codes for your valine. Fine, it codes for your valine. Fine, bache. Nageshwar, bache, it's your choice, right? It's your choice. If you can, that's very good, because yes, there are chances that codon based question can come. But if you cannot, right? So at least you should focus which codons are for the stop codons, right? Which codons are for the initiator codons, right? Leucine is having maximum 6 different codons. Only methionine and tryptophan, they are coded by one codon. So such information you need to remember then. Fine, bache. So GUG can also act like the start codon. So GUG codes for what? GUG codes for your valine. Clear, bache? Now I'll relate one thing here. Do you remember sickle cell anemia? We have discussed that in principles of inheritance and variation. Do you remember sickle cell anemia? Yep, sickle cell anemia. So you know that when you talk about the hemoglobin, it is having 4 chains. Okay, it is having 4 polypeptide chains. How many chains are there? 4 polypeptide chains are there, right? So 2 alpha chains and 2 beta chains. 2 alpha chains and 2 beta chains. That's what we have. So when it comes to the beta chain, so in beta chain at 6th position, in beta chain at 6th position, you know now what is going to happen? Your glutamic acid is replaced by valine. Glutamic acid is replaced by valine, if you remember. Good valine is the trick here. So glutamic, good valine is the trick here. U is there. So glutamic acid will be replaced by valine. Remember, bache, GLU is your glutamic acid. Okay, GLU is your glutamic acid. But if it is GLN, it is your glutamine. So please don't get confused here. GLU is your glutamic acid. GLN is your glutamine. Same way, aspartic acid, ASP, ASN is your asparagine, I'll tell you. Okay, so for glutamic acid it was, the sequence is GAG, for valine it is GUG. So see, there is the difference of only one base, right? There is a difference of only one base. Remember I told you when I was teaching you principles of inheritance and variation, that let me teach you the genetic code, then you can understand these things in a better way. Okay, so just look at this table, see here. See, so for asparagine, it's ASN. Okay, for aspartic acid, it's ASP. For glutamine, it's GLN. For glutamic acid, it's GLU. Okay, it's GLU. So see this isoleucine, methionine, valine. So that's how you can remember. Fine? Fine. So this is about the genetic code. This is about the genetic code. So, okay, I challenge you to remember this genetic code. Can you? I challenge you to remember the genetic code, this complete checkerboard, right? That which codon is coding, which is coding for which amino acid. Can you tell me? Can you? I'm just challenging you. It's not mandatory that you have to accept the challenge. Seriously, challenge accepted. Not accepted. Okay, fine. Seriously. Sure. Sure. Okay. Okay, so now let's talk about the salient features of the genetic code, another important topic. Okay, it is your another important topic. The salient features of what? The salient features of the genetic code. So as I said, the codon, first of all, the codon, this genetic code is universal. There are few exceptions only, right? There are few exceptions only. Otherwise, it is what? It is universal. The codon is triplet. 61 codons code for amino acid and 3 codons do not code for any amino acid. I told you they are stop codons. So again I'm repeating so that you can remember. It's UUU, then AG, then AGA. So these are 3, yes bache, 3 stop codons. Clear? Next, one codon codes for only one amino acid, hence it is unambiguous and specific. Yes, your genetic code is specific. If I'm saying UUU is making phenylalanine, bache, it will make phenylalanine only in every possible situation. It's very specific. There is no ambiguity, right? In genetic code, there is no ambiguity. Ambiguity means there is nothing doubtful. It is very specific. It will code that amino acid only. Okay, so that's why this is the property of the genetic code, that it is unambiguous. Okay, it is unambiguous. It's specific. There is no chance of being doubtful. Okay, then see, these are the two properties where students, they get confused. One is unambiguous nature, and yes, in paper they can even confuse you, right? So they sometimes yeah, I was solving some PYQ, so I checked it. Instead of unambiguous, they have mentioned the ambiguous word, and students were like, yes, we have read something like that in NCERT, and they marked it. Actually, it is incorrect. Your genetic code is unambiguous, or you can also call it as non-ambiguous. Okay, so there are two words. One is unambiguous nature of genetic code, and second is degenerate, or you can say that this property is of degeneracy. As per this, we are saying that these codons are specific. If UUU is coding for phenylalanine, in every possible situation it will code for this only. If GUG is coding for valine only, it will code for this only. But when you are saying degeneracy, when you are talking about the degeneracy, but here we are saying that one amino acid can be coded by, it can be coded by different codons as well. One amino acid can be coded by different codons as well. I'll give you one example. UUU, UUC. They are two different codons, but they are coding for the same amino acid, that is phenylalanine, right? That is phenylalanine. Like, I'll quote one example here. See, if I'm a biology teacher, I will teach you biology only. Am I right? If I'm a biology teacher, right, I'll teach you biology only. Isn't it? It means it's right. So this property is being non-ambiguous. I am biology teacher, I'll teach you biology only. Now, when it comes to the degeneracy, how can I explain? You are my student. Let's say Sabha is attending my classes for biology. Sabha is attending Wasim sir's classes for chemistry. Sabha is attending Shreya sir's classes for the physics. So she is a single student, right? She is, because she is reading 3 different subjects. So she is going to 3 different teachers. She's going to she's following 3 different teachers for 3 different subjects. Okay, but she is the same student. So here also the amino acid is same, but the codons can vary. Clear bache? Okay, so being unambiguous means that these codons are specific. Being degenerate means, right, that different types of codon can code for same amino acids. Okay, okay, so do not get confused here, please. Fine. So see this part. So some amino acids are coded by more than one codon. Some amino acids are coded by more than one codon. So hence the code is degenerate. Done, bache? So codon is read in mRNA in a contiguous fashion. I told you no punctuations are there. First three, next three, next three, like this. It is nearly universal. What is the meaning of being nearly universal? Like only few exceptions are there. Otherwise, in all the organisms, the same, right? This genetic code is same. So like see, from bacteria to humans, UUU code for phenylalanine only. Right, bache? So some exceptions are there in mitochondrial codon and some are in the protozoa. Like in the case of paramecium, you will see some exceptions, and even in the case of mitochondrial codon as well. Clear, bache? So AUG is having the dual function. It is also the initiator codon. Then it is also the initiator codon. Done, bache? Done, bache? Okay. So now here, when you are talking about the degeneracy, you know, this degeneracy is because of the third base. It is because of the third base. It is because of the base which is present at 3' Do you know that? It is because of the base which is present at 3' Right? So that third base is also known as wobbly base. And bache, I'll teach you after. Right? After this, we'll start wobble hypothesis. Then I will tell you about it. So this degeneracy is because of this wobbly base. Clear, bache? So the properties of genetic code, they are very important. So here you guys can see that if following is the sequence of the nucleotide, right, predict the sequence of amino acid. So you know that the first codon will always be your methionine, okay, because it is the initiator codon. Then phenylalanine, 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 phenylalanine. See, okay, got it? Done. So I'll tell you about I already told you about the sickle cell anemia. Now I'll tell you about the frame shift mutation, and then we'll start the translation. Okay, then we'll start what? We will start the translation. So excited? Excited? We are about to finish this chapter today. So mutations, you know that what are mutations? Mutations are the sudden changes. And yes, these sudden changes are heritable as well. Means they can pass from one generation to another. Isn't it? And when you talk about the mutations, first time Hugo de Vries, right, he explained about it. He was the first one, if you remember. He studied the evening primrose plant, that is your Oinothera lamarckiana, and he noticed that progeny is totally different from the parental plants. He observed the progeny of the Oinothera lamarckiana, which is evening primrose, and he observed that progeny is different from, right? It is entirely different from the parent plant. Not all the individuals, not all the offsprings, but yeah, some of them, right? Some of them. Okay. So he gave the concept of that mutation. So mutations are sudden changes which are heritable. If in general I talk about the mutations, but only useful mutations are passed to the next generation. If it is harmful, it is destroyed. Okay. And generally they are recessive, right? Mutations, generally they are recessive. And you know, these mutations are not always harmful. You know that. It is a property of the genetic material that it should allow slow changes. It should allow that slow mutations. Why? Why? Because that will result in variation. And that variations are important for adaptations. Isn't it? That variations are important for adaptation. Isn't it? So that's why even mutations are important. And even, you know, we have the example of induced mutations as well, like where scientists, they used to induce the mutation so that they can get a particular type of, you know, so that they can alter the chromosome number and they can get their desired result, like wheat that we used to have, right? The wheat that we used to have, that is also the result of the mutation. Clear, bache? Clear, bache? So the relationship between genes and DNA, it is best understood by mutation, right? Right. So effect of large deletions and rearrangement in the segment of DNA is easy to comprehend. I told you before, like in the last chapter also, even we talked about the chromosome, that if there is inversion, if there is deletion, we can see that results, isn't it? Isn't it? Even if you are talking about the segment of DNA, if there is any change in that gene, right? You know the example of sickle cell anemia. So sickle cell anemia is the example of point mutation. Point mutations means there is a change of only one base pair. Imagine, right? There is a change of only one base pair. There is a change of only one base pair. Your GAG will become GUG in sickle cell anemia. So it's point mutation where only one base will be changed. Clear, bache? So obviously if there is any change in that gene segment and DNA segment, it will reflect in the protein. It will reflect in the amino acid there. Clear, bache? Clear, bache? And you know, because of that, what was happening? The RBC, they will become sickle shaped, right? Right. So that person can have anemia as well. That person is having the anemia as well. Done. So this is one example of the mutation here, the point mutation. Next is your frame shift mutation. Now see, they have given you one line. It is Ram has red cap. Okay. So if we insert a letter B in between has, obviously it will change the meaning. Same here. Like see, if you have let's say it is like this. See, if the amino acid sequence is like this, so you know that methionine, phenylalanine, phenylalanine, methionine. Let's take the example. But if I add any other base here, let's say if I add A here, right? If I add A here, so it will become AUG for methionine. Now the next base will be AUU, UUU, CAU. So it will change the sequence of my amino acids. It will change the sequence of my amino acid, yes or no? It is going to change the sequence of my amino acid. Of course, yes. Of course, yes. Let's say now I'm adding two bases. After A, again I am adding U. Again I am adding U. Then see, AUG, AUU, UUU, UCA, UG. These two bases, they are coding for nothing now. Again, the reading frame will change. No doubt we have to move from 5' to 3' end, but that reading frame will change. But bache, let's say if I will add three bases, so I know that three extra bases added, they will code for another amino acid. So no doubt in that protein, in that particular protein, one more amino acid will be added. Fine. Okay, so in that way, like if three bases are added or deleted, the damage will be comparatively less. Okay. So see, they are giving one example. Ram has red cap. So if they are adding B here, Ram has BRE, red, like it is changing the meaning. Right now, let's say they are adding two letters here. Ram has BIR EDCAP. Again, it is not making sense. Now let's say they have added three letters and these three letters are making one word. Ram has big red cap. Okay. Okay. So same kind of things. Same kind of things are there in the amino acid, in the nitrogenous bases sequence as well, right? So if there is the insertion or deletion of one or two bases, it can change the reading frame, right? From the point of insertion or deletion. So mutations, such mutations are actually the frame shift mutation, right? Right? These are the frame shift mutations. It can be because of insertion as well, because of deletion as well. Clear, bache? Because of insertion as well, because of deletion as well. Okay, so can I ask you one more question? Can I ask you one more question? Even I'll help you, bache. Can you just tell me that from this codon, which amino acids are you going to get? Right? Can you just tell me? See, you can take the help from this table. Okay, you can take the help from this table. Here you have the sequence. You just need to tell me that this sequence is going to form which amino acids or which, right, what will be there here? Sangeeta bache, no, I don't think so. Baki, let's see bache, if you have to go for something else, right, you can go. You can watch the recording later, okay? So just tell me the sequence in one message. You have to tell me the sequence. Quickly. Quick. You have to tell me the amino acids, that which amino acids will be there in your polypeptide. You can take the help from this table, na? It's easy. It's easy. Here you have the sequence, here you have the table. You just need to tell me about the sequence of amino acids. Anyone? Seriously? I really don't think, bache, if you cannot give the answer of such simple question, then I don't know how to help you people. Very good, Subhashini. Very good, Subhashini. But Subhashini, you are making a mistake here. You are right. When it is CUC, it means leucine, right? And then when it is AUC, it means isoleucine. You are right. Then when it is GUU, it is valine. Yes. It is valine. Yes. And then when it is it is GUU, it is valine. Then UCC means it is serine. Okay, it is serine. Then comes your CCC, it means your proline. Okay, but it is UGA. UGA means stop codon. So if there is any stop codon in this, right? If there is any stop codon here, means you have to stop the process here. The stop codon is indicating that now the translation will terminate, right? There is no need to tell further, Sandhya, that there will be the tyrosine and tyrosine. No, it is not going to happen, Priyanka. Okay, it is not going to happen. So when there is a stop codon means the translation will stop here, right? The further codons will not be translated into amino acids. Okay, it will not be translated into amino acid. So stop codon is saying that, okay, fine, now stop the process from this particular, right? From this particular sequence, these are the amino acids that you people are going to get. These are the amino acids that you people are going to get. Is that clear? Done. So this is all about the genetic code, and next is your tRNA, right? We have already discussed the structure of tRNA yesterday, but still, let's revise it quickly. Okay, let's revise it quickly. We have already covered it in the last class. So hope you know about it. Clear? tRNA. So, bache, when you talk about the tRNA, it is your transfer RNA, it is your adapter RNA, it is your soluble RNA, and it is smallest RNA. So, it is making 10 to 15% of your RNA, right? It is making 10 to 15% of the RNA of your cell. Clear, bache? So, when you talk about its two-dimensional structure, it's a clover leaf-like structure. What kind of structure is there? It is a clover leaf-like structure. Clear, bache? And when it is a 3D structure, it is going to be inverted L, right? It is the inverted L-shaped structure. You need to remember this. Fine, you need to remember this. Now, this is the 5' end, this is the 3' end. Always remember when it is the 3' end. Okay, always remember when it is the 3' end of your, yes, of your tRNA. It is having free OH group, and towards this 3' end there will be a sequence CCA. Always you will see a sequence CCA is present there. Okay, Subhashini, CCA is present there. Now this arm is your DHU arm, DHU loop. This is T psi C, and this is your amino acid acceptor arm. Fine, this is amino acid acceptor arm. This one is your anticodon arm or anticodon loop. This one is variable arm, right? So this one will just help us to categorize the tRNA into two different classes. That's all. So when it is the amino acid acceptor arm, this OH group of the 3' end, it will react with amino acid, bond formation will be there, and this side will pick up the amino acid. And how is it possible? When your tRNA will get charged. And how the tRNA will get charged? when it will bind with an enzyme. Which enzyme it is? It is your amino acyl tRNA synthetase. It is your amino acyl tRNA synthetase. What's that? It's your amino acyl tRNA synthetase. So you have DHU here, dihydrouridine, unusual base is present there. T-Psi-C, the Psi means pseudouridine, another unusual base. This is anticodone loop and this is anticodone arm. So actually it is having the anticodone, mRNA is having codone. So the anticodones of it, they are present here. They are present here, and as per that, this amino acid will, this tRNA will get charged and will pick up the amino acid from the cytoplasm. First point. Now, this D arm, D for down. So when we are down, we need energy. When we are down, we need energy. So we are going to get that energy, that charging from the enzyme. So this particular arm, this DHU arm, is going to bind with the enzyme. You know the name of the enzyme, bache. So when this tRNA, it binds with that enzyme, it will get charged. So what is the meaning of charging of tRNA? It will pick up the amino acid here. The amino acid will get attached here. And now this T-Psi-C, it will bind with ribosome. So that's what you need to remember when you read about the tRNA. Okay, so DHU arm will join with the enzyme, T-Psi-C arm will join with the ribosome, anticodone arm will join with the mRNA, amino acid acceptor arm will join with the amino acid. So one molecule is joining with 4 different things, right? That's why we call it as adapter molecule. Done, bache? That's why we call it as adapter molecule. Is that clear? So we have already covered this part in the last class about the tRNA. If you want to know about it in detail, you can check the last session, okay? So, from the very beginning of the proposition of code, it was clear to Francis Crick that there has to be a mechanism to read the code. Right? Francis Crick, he was clear that there should be a mechanism which can decipher these codes. Isn't it? Isn't it? Right? And also to link to the amino acid, because amino acids have no structure specialities to read the code uniquely. So he postulated, who postulated? Francis Crick postulated that there is the presence of an adapter molecule that would on one hand read the code, on another hand would bind to the specific amino acid. So that anticodone arm, the anticodone loop, bache, the anticodone arm, the anticodone loop of that tRNA, it will read mRNA, it will read that mRNA, and simultaneously it will pick up the particular, right, as per that codone, it will pick up the amino acid from the cytoplasm. Right? So it was postulated by Francis Crick. Okay, it was postulated by Francis Crick, another important MCQ. So tRNA is your soluble RNA. Okay, bache, it is your adapter RNA and you know about its structure as well. So see, this is your mRNA, 5' to 3' end. AGU is written here. AGU is written here. Okay, and here what do you have? UCA, anticodone. A is going to bind with U, G with C, U with A. And here it is UAC and C. Here it is AUG. So you know that AUG is the start codon. What is it? It is the start codon. Clear, bache? It is the start codon. Done? Right? So yes, in the exam, the question can come from this part. It is already a PYQ. Right? So they can ask you the sequence of, they can ask you the codons for serine, they can ask you the codons for tyrosine. Why? Because it is given in NCERT. So that's what you need to remember. Okay, that's what you need to remember. These are the anticodones, no? So these are the codons. You have to read in this direction. AGU, UAC. AGU, serine, UAC, tyrosine. Okay? Okay, so that's a PYQ. Actually, it is a PYQ, so please pay attention here. So tRNA, I told you already, it is having anticodone loop that has bases complementary to the code, right? And it is having amino acid acceptor arm, right, to which it binds to the amino acid. So tRNAs are specific for each amino acid. Now come to this point, right? Now I will teach you about the vowel hypothesis. Achha, first of all, tell me, bache, how many of you find the translation difficult? How many of you find the translation difficult? That, ma'am, yes, it's a difficult process. It's not that easy to remember. Yep, it's not that easy to remember. Quick. Like actually in the case of translation, if the machinery is clear to you, if you know what exactly is the ribosome and how many sites it is going to have, how the structure of tRNA looks like, which part of tRNA is going to bind with which part, then it's easy for you. Then it is very easy for you. Okay, it is very easy for you then. But if you don't know about it, then it is difficult. So try to understand, right? Try to understand the machinery. And if there is any doubt, like Sandhya is saying, ma'am, there is a doubt about the directions. Always remember your synthesis direction, your reading frame is always 5' to 3'. It is always 5' to 3'. So, bache, translation is taking place in your cytoplasm, isn't it? The translation, it will be there in your cytoplasm. If you are talking about the prokaryotes or if you are talking about the eukaryotes, in both the cases actually the translation is there in the cytoplasm. But when it comes to the transcription, in prokaryotes, transcription is there in prokaryotes, transcription is there in cytoplasm. But in eukaryotes, transcription is there in nucleus. But here, in both the cases, it is going to take place here in the cytoplasm. Done, bache? The translation will be there in the cytoplasm. That's what you need to remember. Now, what's the next point here? This is one point. Okay. This is one point. What is the next point here? Here in the cytoplasm, you are going to find the amino acids. What are you going to find? You are going to find the amino acids here in this cytoplasm. Okay. And for that amino acids, you are going to have a specific tRNA. Do you know that? Right? Like if we have 20 amino acid, 20 protein forming amino acid, then it means you have 20 tRNAs. Okay. And you have 20 different tRNAs. See how rich these amino acids and these tRNAs are. For methionine, it is having its own tRNA. Valine is having its own tRNA. Serine is having its own tRNA. Are you getting it? Are you getting it, Sandhya, Nandini? Okay, so that's what you need to remember. One thing. But now one more question is there. I told you that there are total 64 codons, isn't it? That's what I just said. That there are total 64 codons. Out of that, no doubt, 3 codons are the stop codons. Okay, 3 codons are the stop codons. But I also told you, bache, that remaining codons, out of these 61 codons, all 61 codons are coding for 20 amino acids, isn't it? All 61 codons are coding for 20 amino acids. It's just that your methionine and tryptophan, they just have singlet codon. Otherwise, all other amino acids, they have more than one codon. Okay, they have more than one codon. So how is it possible? We have 20 amino acid, we have 20 tRNAs, then, but in totality, codons are 61. Then how will it be justified? Right? How will it be justified? So, bache, here comes the role of your wobble hypothesis. Okay. Right? Now you have to understand this. Right? And here, as per this hypothesis, right? As per this hypothesis, your third base is the wobbly base. Do you know the meaning of that? Yes. Do you know the meaning of that? Actually, here, you know that you have the anticodon, isn't it? First of all, this hypothesis was propounded by Francis Crick. Note down. Right? It was propounded by Francis Crick. This is what you need to note down. Now see, your tRNA is having the anticodon. So obviously that anticodon is also having 3 bases, isn't it? Even that anticodon is also having 3 bases, yes or no? Yes or no? Yes or no? See, right, vowel hypothesis we are discussing now, Francis Crick. So I am telling you that when you talk about the tRNA, right, when you talk about the tRNA, when you look at this part, right, here you will find the anticodon, isn't it? What are you going to find here? You are going to find the anticodon here. So no doubt, this tRNA, it is not just going to recognize only one anticodon. It can recognize more than one anticodon as well, right? Actually there is a difference of third base. First two bases, right? First two bases, they follow Francis and Crick. They follow Watson and Crick pairing. Do you know that? First two bases, they follow Watson and Crick pairing. It means A is complementary to U, C is complementary to G, right? First two bases are going to follow this thing. Fine. First two bases are going to follow this thing, but the third base is the wobbling base, right? Right? Third base is the wobbling base. So actually in tRNA, one anticodon can recognize more than one codon. Why? And how is it possible? Because of the third base. Because this third base is not following this Watson and Crick pairing like this, right? It can pair with another bases as well, right, bache? It can pair with another bases as well, right, bache? I'll give you one example, like anticodon AAG. According to you, it should recognize this. AAG is the anticodon. Bache, AAG is the anticodon. So if it is the anticodon, so according to you, it should recognize this. Yes, it should recognize this. Yes or no? Yes or no? Yes or no? But it can also recognize UUU. Do you know that? Do you know that? See, AAG is the anticodon present on your tRNA. This anticodon should, if you follow Watson and Crick pairing, it should recognize UUC. Yes, it is going to recognize UUC, but along with UUC, it can also recognize UUU, right? It can also recognize UUU, right? Why? Because of the change in this third base. So first two bases are definitely following the Watson and Crick pairing. So this anticodon is okay with it, that at least first two bases are following that Watson and Crick pairing. In the case of third base, it is the wobbly base, right? And that is why the property of degeneracy is also there. Are you getting it? Are you getting it? And because of this thing, right, because of this property of tRNA, right, we can say that we have only 20 amino acids, but that 20 amino acids can recognize, right, that 20 amino acids having 20 anticodon can recognize another codons as well present on mRNA. Done, bache? Right, bache? So that's what you need to remember. Is that clear? Is that clear? Yes or no? Done. So the next topic that we need to start is, now the next topic that we need to start is translation. So it is the process of polymerization of amino acids to form a polypeptide. So what are we going to study here, Trisha? That how these amino acids, they are polymerized to form the polypeptide. But before that, let me ask you a few questions, and trust me, you are going to find this topic easy. It is easy. Okay, you just need to remember it in a sequence, like a story, right? If you remember it like a story, then it is going to be easy for you. Okay, then it is, no doubt, it is going to be very easy for you. Fine. So, done. So let's start the translation. Okay, let's start the translation first. So if you want to understand the translation, the machinery required is your ribosome, of course, your mRNA, right? It is your ribosome, it is mRNA, the tRNA, amino acids, okay, and enzyme, of course, right? ATPs, of course, and there are certain factors as well. Fine, there are certain factors as well. The translation factors are there. Okay, certain translation factors are there, ATP and all that things are present, right, bache? This is the first point. Now what's the second point? Let's talk about the amino acid first. I don't know whether you remember it or not. Do you remember this is the basic structure of the amino acid? Do you remember it is the basic structure of the amino acid, having amino terminal, having carboxyl terminal, right? One side is having hydrogen. Here it is R is the alkyl group, and here it can be hydrogen as well. Like in the case of glycine, here you will find H, right? That is the simplest amino acid, glycine. Otherwise, this is the basic structure of the amino acid. This is the basic structure of the amino acid. Clear? Yes or no? It is the basic structure of the amino acid. I hope you know that. Now let's take another amino acid. Right, this is the another amino acid. Yes, bache. So actually what is happening, you know that in the polypeptide chain we need to join, okay, we need to join the different amino acids. And how the different amino acids will join, bache? They will join with the help of peptide bond. How will they join? They will join with the help of peptide bond. Are you getting it? So that peptide bond is actually CONH, right? It is CONH. So see this, it is the carboxylic acid, it is the amino group. So what is going to happen here, bache? Obviously the water will go out, the dehydration will occur, isn't it? The dehydration will occur, isn't it? Isn't it? So you are left with this bond, na? C double bond O, then N, then H. Yes or no? C double bond O, then N and H, and here you will have your that particular amino acid. So bache, that's the peptide bond. So here in this translation, this is going to happen. Okay, here in this translation, this is going to happen. So this peptide bond which is CONH, it is formed by the dehydration of water, no doubt. So here in this translation, when we will discuss this process, I'll tell you that this bond formation is catalyzed by a ribozyme, your peptidal transferase, right? The peptidal transferase, the ribozyme, it's a ribozyme. Ribozyme means RNA is acting like an enzyme. Okay, what is the meaning of ribozyme? It means it is the RNA, right? It means it is the RNA which is acting like an, right? It is the RNA which is acting like an enzyme. Clear, bache? Clear, bache? Right? So peptidal transferase is going to catalyze this bond formation here. No doubt I will discuss the process, but I'm just, telling you the points where you can get confused, or from where the question can come. Okay, so peptidal transferase is the enzyme which will catalyze the bond formation during this polypeptide synthesis. Fine, bache? Clear? So now you know the basic structure of amino acid. So remember, when we will start the charging of tRNA, so during that charging of tRNA, the 3H end is having OH group. The 3H end is having OH group. So to this OH group, right, from this amino acid, this COOH will join, right? Right? So carboxylic group of the amino acid is going to form the bond here. Amino-acyl bond will form here, and then there will be the charging of this tRNA. Right? These are few points where you people get confused most of the time. Clear, bache? Sure? Sure. And now when you talk about the ribosome, let's discuss the ribosomes here. So you know that in the case of prokaryotes, right? You know that in the case of prokaryotes, the story is you have 50S, you have 30S, right? Right. But in the case of eukaryotes, the story is 60S and 40S. Right, 60S and 40S. So this one is the larger subunit here. This one is the smaller subunit here. Right. So bache, always remember that these ribosomes, one is having the larger subunit, another is the smaller subunit. Bache, they do not stay connected to each other. Right. They do not stay connected to each other. This is what you need to remember. Okay, whenever there is the requirement for the protein synthesis, whenever there is a need to synthesize the protein, only then these two subunits join. Do you know that? Otherwise, they are separated. Otherwise, they are separated. So magnesium ion, Mg2 positive, magnesium ion is going to join them. Okay, magnesium ion is going to join them. And remember now, 50S is having your 5S, it is having your 23S ribosomal RNA, this is having your 16S ribosomal RNA. I hope you remember, along with RNA, proteins are also there. Here you are having your 28S, 5.8S, 5S ribosomal RNA, and here it is having your 18S ribosomal RNA. Remember? So obviously in the translation also the basic process is same, but yeah, there are certain differences in prokaryotic and eukaryotic translation. There is a difference of factors as well. Like in the case of transcription, we were saying that prokaryotes, they are having sigma and rho factor, but that factors were absent in the eukaryotic transcription. Here also you are going to talk about certain factors, right? You are going to talk about certain translation factors, especially the elongation factors. Okay, like in prokaryotes, elongation factor TU, TSG is there. In eukaryotes, that will be eukaryotic elongation factor 1, 2, 3, like this. So factors are going to be different. Okay, some joinings can be different a bit, but ultimately the basic process is same. Fine, basic process is same. And yes, moreover, the mRNA is also different now. In the case of prokaryotic mRNA, there is no capping and tailing, right? But in the case of eukaryotic mRNA, you will see 7-methyl guanosine triphosphate cap, and you will see polyadenylated tail. But no such structure is going to be there in eukaryotic mRNA. So these are few points that you should notice. Okay, Nandini? So as I said, they are going to stay separated. Okay, they will join only when the protein synthesis is required. So remember, in your larger subunit, right, you will see some sites as well in ribosome. And these sites are your E, P and A, right? E, P and A. Do you know that? In your ribosome, there are going to be certain sites. Site E, site P, site A. So that's why I'm saying, firstly, understand the machinery, bache, then the process will be easy for you. Now you know the basic structure of amino acid. Now you know the structure of tRNA. Now you know that the carboxylic group of amino acid will form the bond with the tRNA. Now you know that tRNA is having 4 different arms and they will join with 4 different parts. Now you know the mRNA, how they are different in prokaryotes and eukaryotes. And now we are talking about the ribosome, the larger and smaller subunit. It will stay apart from each other. It will only join when there is the protein synthesis, and your magnesium ion is going to help there. Okay, your magnesium ion is going to help there. And when you are talking about this larger subunit, it is having few sites. Site EPA, it's EPA. E for exit, right? P for peptidyl transferase. And A for amino acyl site. Are you getting it? And here, in this ribosome, you will also see a groove-like structure. So when the protein synthesis will continue from this groove, that polypeptide will come out, right? That polypeptide will come out from that groove. Are you getting it, Sabha? The polypeptide will come out from that groove. Is that clear? So E, P and A. E, P and A. So from this groove, polypeptide will come out. From this E side, from this exit side, your tRNA without amino acid will come out. Once tRNA will drop its amino acid, it will come out from this E side, right? So sites, you need to remember that. And actually how that process is going to take place, this is your mRNA, right? To your mRNA, smaller subunit will bind, and then your tRNA will come, and then your larger subunit will come. That's how it works, right? I'm repeating this again, bache. This is your mRNA. To this mRNA, smaller subunit will join, then your tRNA will join, and then comes your larger subunit. So that's how the process starts, right? That's how the process starts, especially in your prokaryotes. So any doubt related to machinery? Yes, bache? Do you have any doubt related to machinery? And remember, this 23S is your ribozyme. It is your peptidyl transferase. It is your peptidyl transferase. So, bache, if there is any doubt related to machinery, tell me, because now I will start the process, and one by one, you have to understand the steps there. Right? You have to understand the steps there. And yes, magnesium ion is very, very, very important for this process. Okay. Magnesium ion is very important for this process. Done? Sure? Okay. So see, EPA site, and that's how it is present. Okay. So ultimately the process is same, bache. The initiation will be there, right? Then the elongation and then the termination, right? The basic process is going to be same. The initiation will be there, the elongation will be there, and finally the termination will be there. And as I said, there are certain factors which are required for this process. Okay, there are certain factors which are required for this process. So when you talk about the translation, so ultimately the first step is going to be the activation of amino acids. Okay, the first step is going to be the activation of amino acid. This is your first step. Fine, this is your first step, that is activation of amino acid. What is it? What is it, Ashar? It is the activation of amino acid. Okay, so now you know your amino acids are there in the cytoplasm, and as for the information given on mRNA, one by one that amino acids will be picked and they will be added to the chain. This point clear? What is happening exactly? We know in the cytoplasm amino acids are there. tRNA will pick it up. As per the information written on mRNA, tRNA is going to pick it up. That's what we know, and that's simple as well. That's simple as well. Now the point here is that how is it possible that directly amino acid will pick up and come? No, its charging is required. Charging means its activation is required. So we have to activate our amino acid as well. We have to activate that particular tRNA as well. As I said, for tRNA and for amino acid, their activation or charging is required, and for charging of the both, for the amino acid as well, for the tRNA as well, the same enzyme is going to play the role. Okay, the same enzyme is going to play the role. So the very first step is your activation of amino acid. And how is it possible? Let's say here you have your amino acid, right, in the presence of and you need the ATP here, and in the presence of enzyme. You know which enzyme it is. It's tRNA amino acyl synthetase, and here you will get 20 different enzymes. Like we have 20 different tRNAs, same way we have 20 different enzymes as well. Clear, bache? We have 20 different enzymes as well. And yes, you also need the magnesium ion. You also need the magnesium ion. So what is happening here? What is happening here? You have amino acid. It will join with AMP. Actually, here it is the ATP, no? So ATP, 2 phosphates will be released, one will join here. And when the 2 phosphates will release, they will provide the energy. So ultimately it is amino acid, AMP, enzyme complex, right? And 2 pyrophosphates, 2 phosphates will be released. Let me explain this part. Let's say that's your enzyme. Let's say that's your enzyme. You know which enzyme it is? It is your tRNA amino acyl synthetase. It is your tRNA amino acyl synthetase. That's the enzyme, tRNA amino acyl synthetase. So with the help of ATP, right, that ATP will provide energy, right? So this enzyme will bind with your amino acid. It will bind with your amino acid, and obviously, right, one AMP is also there. AMP is also there. That's how it is going to do the job. So basically, enzyme is just like something which is going to connect tRNA and amino acid. Like, I hope you know about the wingman, right? Or you can say that a kind of mediator in between. A mediator is conducting, right? Mediator is fixing the meetings, right? Two parties are coming, then they are having their deal and all, right? Same is the case here. So first step is charging of amino acid. Your amino acid will join with this AMP, right? And obviously two phosphates are released, right? It is providing the energy, and then this charged amino acid is going to bind with the enzyme, right? Now the same enzyme, bacha, the same enzyme, it is having the sites for the tRNA as well, right? The same enzyme is having the site for the tRNA as well. Are you getting it? It is having the site for the tRNA. So can you tell me which arm of this tRNA will join with this enzyme? Can you tell me the name of the arm? Yes. Can you just tell me? Because that's the second step. That's the second step. That is your charging of tRNA or loading of tRNA or activation of tRNA. That's your second step here. Okay. Right. So here also amino acyl. This AA means amino acyl. Let me write it. Why amino acyl? Because we are talking about the bond formation here. Amino acyl AMP enzyme complex. Yes. Amino acyl AMP enzyme complex. And with its specific tRNA. Okay, it is going to join here, of course. It is going to join here. So of course it is it will join it with join here with its specific tRNA. So now what will happen? tRNA and amino acid will join. AMP will be released and the enzyme will be released. Okay. Okay, and the enzyme will be released. Clear, bache? Or you can simply write it like this. Amino acyl tRNA. So if I'm using the word amino acyl tRNA, it is indicating that my tRNA is charged. It is indicating that my tRNA is charged. Are you getting it? So I'm repeating this part again, right? ATP is providing the energy. Actually, this amino acid is going to, right, join with that ATP. How? With the help of this enzyme. Actually, what is happening? Amino acid and AMP, they will join. And we are using the word here, amino acyl. Because bond formation will occur, obviously, AMP and amino acid forming bond. The combined structure is amino acyl AMP, and plus they will get attached to the enzyme. So this enzyme is also having the site where the tRNA can bind. So tRNA with the help of its DHU loop will join with this enzyme. So enzyme is like a mediator. Like let's say there is a couple, they are having a fight, right? You are the common friend, right? You are calling them. You are calling them ki you please come to my place, you please come to my place. You are acting like a mediator there, and you are telling them to have that patch up. Okay, that please don't break up, talk to yourself and be a team again. Same is the case here. So enzyme is bringing that charged amino acid and the tRNA closer, and then finally what is going to happen, right? There will be the patch up. So what will happen? The tRNA will join with amino acid, then they both will say bye bye to AMP and enzyme. Right? They both will say bye-bye to AMP and enzyme. I hope you all have done it, है ना? In the case of couple fight, if you are the common friend, right? If you are the common friend, you will always try to fix the things. But at the end, they are going to blame you only. They will be together and then they will say bye-bye to you. Same is the case here. So enzyme will bring them closer. And later on, what is going to happen? tRNA and amino acid, they will join and they will say bye-bye to AMP and enzyme. Okay? They will say bye-bye to AMP and enzyme. So from these two reactions, that's what you guys can understand. Right? Right? That's what you guys can understand. And of course, for these steps, you need your magnesium ion as well. Right? You need your magnesium ion as well. Clear, bache? Clear, bache? So first two steps are charging or activation of amino acid, charging of tRNA, and now there will be the formation of polypeptide chain. Now actually there will be the formation of polypeptide chain, right? So when there is the formation of polypeptide chain, of course the first step is here, it will be chain initiation. Firstly initiation will occur, then your elongation, and finally the termination. Any doubt? Any doubt? है ना? Trisha, Kaustuba, that's our life story, so you can relate it. You can relate it here. Okay? Okay, okay, okay. Done? Okay. So now what is happening? Let's talk about the prokaryotes. Actually, bache, when it is the initiation, you need, of course, mRNA. You need charged tRNA. Now, what is the meaning of charged tRNA? It's the loaded tRNA. It is the one which is having the amino acid. So whenever we will use the word amino acyl tRNA, so you have to understand that it means it is the charged tRNA. Clear, bache? So mRNA, charged tRNA, GTP, certain initiation factors you need here. Okay, right? So mRNA, charged tRNA, GTP, and you need certain initiation factors. Clear, bache? Certain initiation factors are required here. So IF1, IF2, and IF3 in the case of prokaryotes. In the case of eukaryotes, these initiation factors are different. But in the case of prokaryotes, that's what you need. Right? What you need for initiation, bacha? You need mRNA, you need charged tRNA, you need GTP, you need the initiation factors. Okay, you need the initiation factors. So initiation factors are nothing but they are the proteins. They are the proteins. So for prokaryotes, it's IF1, IF2, IF3. Okay, for eukaryotes, it is something like that, EIF, initiation, eukaryotic initiation factor. But go as per your syllabus. So IF1, IF2, IF3. Clear? IF1, IF2, IF3. Clear, bache? Clear, bache? That's what you need. Now, your GTP and initiation factors are important for initiation. I'll tell you how. Firstly, let's talk about the mRNA. So one by one, please try to understand it. Right? Try to understand each and every step here. It's easy. Trust me, it's easy. This is your mRNA, bache. You have 5' end, you have 3' end. I am talking about the prokaryotic mRNA. No doubt, bache, this mRNA is going to have the initiator codon. Right? It is going to have the initiator codon. And you know that which codon it is? It is AUG. Right? That is going to code for methionine. That is going to code for methionine. But in prokaryotes, actually it is formyl methionine. But in prokaryotes, what's that, bache? It's actually the formyl methionine. It is what? It is the formyl methionine. This methionine is formylated. Okay, this is formyl methionine. Is that clear? Now, what is going to happen here? Few bases above, right? Few bases upstream to this initiator codon. Let's say this is AUG, right? Here you are going to find a sequence. You are going to find a sequence of bases. Do you know that? Here you are going to find a sequence, bache. And what is the name of that sequence? That sequence is your Shine-Delgado sequence. Shine-Delgado sequence. What's that, bache? That is your Yeshiva. That is your Shine-Delgado sequence. Clear, bache? Are you getting it? I'm talking about the prokaryotes as of now, not the eukaryotes, because in the case of eukaryotes, the story is little different. The story is different. So are you getting it? Yes or no? So in the case of mRNA, in the case of prokaryotic mRNA, okay, so you have, right, just upstream to the start codon, you are going to get a sequence. Okay, you are going to get a sequence. The sequence is also of near about 8 nitrogenous bases. Right, it is the sequence of near about 8 nitrogenous bases. So the name of the sequence is Shine-Delgado sequence. So in prokaryotes, this is the sequence that you will find, Shine-Delgado sequence. So this sequence helps mRNA to get attached to smaller subunit of ribosome. Are you getting it? To get attached to smaller subunit of ribosome. Is it clear? So basically, if this is prokaryotic, mRNA, it will join with the 30th smaller subunit, right? How? Because here it is having the Shine-Dalgarno sequence, right? It is having what? It is having your SD. For short form, it is SD, Shine-Dalgarno sequence. So your 30S ribosome, that 60S ribosomal RNA, it is having complementary anti-Shine-Dalgarno sequence, and they will join. Your 30S smaller subunit is made up of 16S, right? It is made up of 16S ribosomal RNA, so it is having anti-Shine-Dalgarno sequence, and that's how their binding will occur. The first step clear, right? The first step clear. And bache, here which factors do you need, right? For this step, you need your initiation factor 3 and GTP is required, right? IF3 and GTP is required for this process. And always remember, magnesium ion is very important for translation, because it is the one which will help the smaller subunit to get bind to the larger subunit. And it is also helping in the charging of amino acid. So that's important. This ion is important. So first step clear, right? So what happened first? Activation of amino acid, then activation of tRNA. For both these steps, only one enzyme was responsible. The same enzyme charged the amino acid, the same enzyme charged the tRNA. What is the name of that enzyme? It is amino acyl tRNA synthetase. So when I'm using the word amino acyl tRNA, means I'm talking about the SABA, I'm talking about the charged tRNA. I'm talking about the charged tRNA. So after the charging part, what is going to happen? The polypeptide formation. What is going to happen? The polypeptide formation. So initiation, elongation, termination, these are the 3 steps here. So when you start the initiation, what you have to do? You have to join your mRNA with the smaller subunit. Okay, and for that we need we need initiation factors, we need GTP. Right, we need guanosine triphosphate. So what is going to happen here, bache? The mRNA of the prokaryotes, it is having a sequence known as Shine-Dalgarno sequence. This sequence will be recognized by the smaller subunit of ribosome, right? This smaller subunit of ribosome is having the complementary sequence, anti-Shine-Dalgarno sequence, so they both will join. And for their joining, initiation factor 3 and GTP and magnesium ions, they are playing the role. So the first step clear here. Yes, first step clear. Now, if I talk about the eukaryotes, in the case of eukaryote students, you know that they are having 7-methyl guanosine triphosphate cap. 7-methyl guanosine triphosphate cap. So that cap helps the mRNA to bind with the smaller subunit. Clear? Clear, bache? So in eukaryotes, smaller subunit of ribosome will be recognized by 7-methyl guanosine cap. Is that clear? By 7-methyl guanosine cap. Okay? Okay? Sure? Now, what should be the next step? Now see, they join here. Somewhere here you are having your initiator codon, right? They joined somewhere here, they are having their initiator codon. Are you getting it? They are having what? Let's say they joined with the help of that sequence. Here you are having that initiator codon, AUG, and you know that Kostuba, because it is a prokaryote, it is going to be formyl methionine. Okay, it's a prokaryote, so here it is formyl methionine. What is it? It is formyl methionine. Is that clear? Is that clear? Yes or no? What is it? It is formyl methionine. Now, bache, your charged tRNA will come, right? Your charged tRNA having the anticodon, it will come here. It will come, and now your charged tRNA will bind to this complex. Okay. Your charged tRNA is going to bind here, right? And this step, let's say if I'm naming this as first and this as second, so for this step also you need GTP, you need initiation factor 1 and initiation factor 2. Are you getting it? Are you getting it? So tRNA will come, will recognize the codon here. It is having that anticodon, so they are going to bind. They are going to bind, and this step also require your, yes, bache, initiation factors, GTP, and of course your magnesium ion as well, right? Of course the magnesium ion as well. Yes or no? Yes or no? Now do you know what is going to happen? And take a count of GTP also. For initiation, you need 1 GTP here. You need 1 GTP here. Okay. Okay. And now what is going to happen after this, bache? And yes, don't forget magnesium ions. Now, bache, your ribosome, larger subunit of ribosome will come. Right? What is going to happen now? The larger subunit of ribosome will come. And let me tell you, once this larger subunit join here, your initiation factors will say bye-bye because their job is done. Okay? Once the larger subunit join to this setup, right, the initiation factors will leave it. And here, this first tRNA, do you know? It is present at which site? At P site. It is present at P site. Clear, bache? It is present at which site? P site. P site. So next to it is A. This is going to be the exit site. So once they join, okay, Subhashini, once they join, initiation factors will leave. Okay, initiation factors, they will leave. So here you know that the 5' end, here you know that the 3' end. Any doubt? Any doubt? Yes. Bache, up to this part, if there is any doubt, do let me know. Do let me know. Okay, so E for exit site, P for peptidal site, A for amino acyl site. Amino acyl site. These are the 3 sites here. Clear, bache? These are the 3 sites here. So any doubt here? If there is any doubt, do let me know, bache. Sure? Sure? Can we talk about the next part now? Yes, can we talk about the next part now? So next part is nothing, but it is what? It is your elongation. Next part is what, bache? It is your elongation. Clear? Clear, clear, clear? It is your elongation. So remember one thing. When it is the translation, bache, when it is the translation, actually what is happening? Your mRNA will not move, but your ribosome will move. Right? It is your ribosome that will move, and it will move here on in this 5' to 3' direction. Okay, like let's say after this particular step, I told you your formyl methionine. Okay, your charged tRNA is present at P site, your first charged tRNA. Now second charged tRNA will come to the A site. Right? Second charged tRNA will come to the A site. Yes or no? Yes or no? And it will come in this way that these 2 amino acids, they will face each other. Right? Why? Because they want to form the bond here. Okay. They want to form the bond here. Let me magnify this part. Let me... So, bache, here the visualization is very important. So, let's say this is a story. This is a tRNA at P site. Now, next tRNA, next charged tRNA is coming to your A site. So, just to show you this part, I'm drawing it like this. Now, these 2 amino acids, what will they do? They will form the bond. What will they do? They will form the bond. Yes or no? These 2 amino acids, they will form the bond. Yes or no? Yes or no? Peptide bond will form here. See, this first amino acid is at P site. The next charged amino acid is at A site. Right? The next charged tRNA is at A site. Now, the amino acids present on these tRNA, they will form the bond. Nandini, they will form the bond. They will form which bond? They will form peptide bond. And you know that I told you already, this peptide bond, its formation is catalyzed by peptidal transferase, and that peptidal transferase is your ribozyme. So actually in prokaryotes, 23S ribosomal RNA, 23S ribosomal RNA is catalyzing this part. It is catalyzing this part. Is it clear? It is catalyzing this part. So after this, what is going to happen? Let me show you. So this amino acid will come to this part. to your second amino acid. Now you will get the dipeptide. Two amino acids will join here, you will get the dipeptide. Right, now this amino acid is all free. It is all free. It will pick up another amino acid. Is that clear? This tRNA is totally free. It will pick up another amino acid. This step is going to take place. Now, after this step, your E, P and A. See students, compare. First amino acid is at P side, first tRNA, charged tRNA is at P side, second charged tRNA is at A side. Now, after forming the bond, that bond formation is catalyzed by peptidyl transferase, which is a ribozyme. Your ribosome will again slide. Obviously, it needs energy for this. It needs factor for this, because the step now that we are discussing is your elongation. Right, it's the next step of the polypeptide formation. It is elongation. So there also certain factors are going to play the role. Are you getting it? Certain factors are also going to play the role here. Are you getting it? Yes or no? Yes or no? Yes or no? Quickly tell me. And how is it possible? How is it possible? It is possible. See, bond formation I told you already. Now I'm saying that your ribosome will slide, right? And for the sliding of ribosome, what is going to happen? Ribosome slide in 5' to 3' direction, right? It is sliding in 5' to 3' direction, and it needs GTP for it, right? It needs GTP for it, of course, and one enzyme is going to play the role here, bache, that is translocase. Actually, it is translocation now. Your ribosome is sliding now. It is translocation now. So, translocase enzyme is playing the role here. It is the translocase enzyme which help in the movement of ribosome, and GTP is going to provide energy for it. So it's not like that after formation of dipeptide that P will automatically come to E. No, it's not like that P will move to E. No, it's your ribosome that will move. It's your ribosome that will slide. So because of that, the tRNA which used to be at P side, it comes to the A side. The tRNA which was at P side, it will come to E side. The tRNA which was at A side, it will come to P side. Now, are you getting it or not? Are you getting it or not? Yes? Are you getting it or not? Tell me quickly. Huh? Are you getting it or not? Of course it is. So it is your ribosome which will slide. So for the sliding, GTP will give you the energy, GTP will provide the energy, translocase enzyme will play the role. So when it will slide, the amino acid, the tRNA at A will come to P, the tRNA at P will come to E, and then it will leave. E means exit, it is the exit side. So this free tRNA will leave. And now the next charged tRNA will come to A side. It will come to A side. So that's how the elongation will occur. Okay, that's how, right? That's how the elongation will occur. Fine, are you getting it? So again the ribosome will slide. Again here the bond formation will occur. Again the ribosome will slide, bache. And for that sliding, GTP will provide the energy, right? And your translocase will play the role. Are you getting it? Your translocation will play the role. So that's how it will move. Clear, bache? And in prokaryotes, remember, you have elongation factor 2TUS-G. 2TUS-G. These are the elongation factors in the case of prokaryotes. Right? In prokaryotes, these are the elongation factors that you have. Done, bache? These are the elongation factors that you have. In the case of what? In the case of prokaryotes. Fine, bache? In the case of prokaryotes. So this process will continue. The elongation will be there, and the amino acids will be further added to polypeptide chain. So once they will find the stop codon here, let's say here it is UGA. so it will stop the translation. Clear? If it will find the stop codon here, that will stop the translation, bache. And for stopping that translation, release factors are going to play the role. Okay, like release factor 1, release factor 2 and 3, they will play the role here. They will release that polypeptide, and from the groove here, this polypeptide will be free. Clear, bache? It will be free. Clear, bache? Yes. Right? So when they will find the nonsense codon, the stop codon, the translation will stop. And I told you 3 release factors are going to help here. Your release factor 1, 2 and 3. Done, bache? Done, bache? Done? Sure? Are you sure? So release factor 1, 2 and 3 will play a role here. And remember, here also GTP is required. So obviously protein synthesis is an energy taking process, right? It is energetically expensive. It is also energetically expensive. That's all. This is actually simple. Okay. And after the stop codon here, you will find untranslated region. Before the start codon here also, let's say you will find untranslated regions. So it is given in NCERT, and even in last class I showed you this UTRs, untranslated regions are there. Untranslated regions are there, and they will make sure the efficient translation actually. So these are the points, these are the processes that you need to remember. So here you guys can see the wonderful diagrams. So see, this is the larger subunit, this is the smaller one, and here it is the P side. So next charge tRNA will come to the A side. So it is as per the sequence present on mRNA. Okay, so see, initiator codon, this is AUG, so it is coming, it will move to this, right? So initiator codon is AUG, its anticodon is UAC, so that's how it will bind. So I'll share the PDF so that you can access these images and you can see it wonderfully. So see, the ribosome is sliding, it will find its initiator codon, right? It will find its initiator codon, they will bind, and then what is going to happen? Larger subunit will come, right? So see, A side, AUG, right? So that's how they are coming. Clear, bache? Clear, bache? Okay, so this is the growing polypeptide as well. Next will come to A side. Okay, so finally polypeptide will be released from the groove present in RNA, and that's the different structures. So see, tRNA is coming out from exit side, 2 amino acids, they are forming the bond. So when they are forming the bond, this polypeptide, this is the groove here, this polypeptide will be released from this part. Okay, and see the next amino acids, they are coming to the A site again. Next amino acid, they are coming to the A sites again. Done? Sure? Are you sure? Yes? So remember, elongation factors are there. So it is the complete process, initiation, elongation and termination, right? So this is the complete process here. So you can just have a look, right? So read it, have a look. And if there is any doubt, do let me know. After this, we'll do the NCERT reading and then we will start the regulation of the gene expression. Fine. After it, we will start the regulation of the gene expression. That is your operon and all. Today we can finish this chapter. Okay, today we can finish this chapter. Done? Any doubt? Sure? So let's read it from NCERT and then we will start the next part. Okay, only 3 topics are left and then we will end the class. Fine, only 3 topics are left and then we will end it. So here you guys can see that when you are talking about the translation, right? When you are talking about the translation, it is the process of polymerization of amino acid to form a polypeptide. So the order and sequence of amino acids is defined by the sequence of bases in mRNA. So there, the mRNA, right? There is a reading frame. In 5' to 3' direction, your ribosome will move. It will read the codon present on mRNA, and then obviously accordingly amino acids will be added. Okay, accordingly, the amino acids will be added. Yes or no? Yes or no? Yes, bache. Yes or no? Right? So the amino acids are joined by a bond which is known as peptide bond. So formation of peptide bond requires energy. Okay, it requires energy. So in the first phase, what will happen? Amino acids are activated. So it is a previous year question. They have asked you that in the translation, which step is the first step? Charging of tRNA or charging of amino acid or initiation? So you have to remember, right? It is the charging of amino acid which is the first step. Clear, bache? And then comes the charging of tRNA as well. Okay, and it is also known as amyloacylation of tRNA. So charging of tRNA, aminoacylation of tRNA is again same. Okay, it is again same. So if two such charged tRNAs are brought close enough, the peptide bond will form between the amino acids, of course. Right? See, if they will be close enough, the peptide bond will form, of course. Right, bache? Right, bache? Right? So they have mentioned the presence of a catalyst would enhance the rate of production. So they didn't mention the catalyst here, but you know, right? But you know the name of the catalyst. That is your peptidyl transferase, which is a ribozyme. So in prokaryotes, it is your 23S ribosomal RNA, which is acting like this ribozyme. Okay, Sabha. In prokaryotes, it is your 23S ribosomal RNA, which is your peptidyl transferase. So cellular factory responsible for synthesizing protein is ribosome. Of course, we know that. That's why we used to call it as protein factory, isn't it? That's why we used to call it as protein factory. So ribosome is having structural RNAs and about 80 different proteins in its inactive state. So you remember this question was there in NEET 2023 and many students were saying that it is out of NCERT, but it is not. Okay, it is not. They were asking this na, this question was there na in NEET 2023. So ribosome consists of structural RNAs, ribosomal RNAs and about 80 different proteins. So it's a previous year question, Sabha. It's a previous year question. So it is in its inactive state, it exists as two subunits, a large and a smaller one. So when the small subunit encounters an mRNA, means when small subunits join with mRNA, the process of translation will start. So there are two sites in the large subunit, bache, for subsequent amino acid to bind and to be close enough to each other for the formation of peptide bond. So obviously here they are talking about the P and the A site. So this process is not wonderfully explained in NCERT, but I have given you the idea, bache. And after all that, after reading that notes, you can just read it from the NCERT. Okay, so NCERT will be easy then. Fine, NCERT will be easy then. So ribosome, as I said, they are acting like a catalyst. See, 23S ribosomal RNA in prokaryotes, which is ribozyme. Done, which is ribozyme. Another important MCQ. Another important MCQ. Done, bache. So a translational unit in mRNA is the sequence of RNA that is flanked by the start codon and stop codon. So ultimately, the sequence which is there in between start and stop codon will be translated. But along with that, as I said, untranslated regions, UTRs, are also present. So they are present at 5' end before start codon and 3' end after start codon. Clear, bache? 3' end after start codon. So for initiation, ribosome binds to mRNA at the start codon, and that is recognized only by initiator tRNA. So ribosome proceeds to elongation phase of protein synthesis, and finally what is going to happen, bache? Complex composed of an amino acid linked to tRNA will come, and that's how the process go on. And here, see, this in the NCERT also, they have mentioned the release factors which are required at the end. So the release factors that I have mentioned, RF1, RF2, RF3, it's in the case of prokaryotes. It's in the case of prokaryotes. Done? It is in the case of prokaryotes. Done, bache? So next topic is your gene expression and then the human genome project and DNA fingerprinting, and the chapter will be over. So that new topic which is given in your syllabus, protein biosynthesis, I will discuss that too, but in a separate lecture. Okay, in a separate lecture. Done. So now I'll give you a break till 1:45. Okay, and then we will continue this part. Fine, so you have 15 minutes. Done? You have 15 minutes. So there will be a break till 1:45 and then we will continue this portion. Okay? So take care. Bye-bye. See you all at 1:45. Hi everyone, welcome back. Hi, welcome back. Okay, so now we need to start the regulation of the gene expression. See, how a gene is going to express itself? Of course, the gene which is present on DNA, that gene will form mRNA and then it is going to form the protein, isn't it? It is going to form the protein. So gene is going to express itself by forming protein. That's what we know. So but this expression can be controlled. It can be controlled at different levels. Like if I talk about the prokaryotes, because you know that in their case, the formation of mRNA and the protein synthesis is simultaneous, isn't it? In the case of prokaryotes, it is the cytoplasm where the transcription will occur. It is the cytoplasm where the protein synthesis will occur. So it's a simultaneous process. Okay. So what is going to happen there? There is the formation of the polysomes there. Okay. There is the formation of the polysomes there. Isn't it? Isn't it? So if you talk about the prokaryotes, if you want to control the gene expression in their case, so you can do it during transcription. Okay. It is the transcription part where we can, regulate that. After transcription directly it will go for the protein synthesis. So at that level we can regulate it. But in the case of eukaryotes, there are different levels. So have a look, bache. So here you guys can see that regulation of gene expression, it refers to a very broad term that may occur at various levels. So considering that gene expression results in the formation of polypeptide, it can be regulated at several levels in eukaryotes. So in eukaryotes, at the level of transcription, okay, because you will have that primary transcript, you will have that heterogeneous nuclear RNA first. Yes or no? What will you have there? You will have your heterogeneous nuclear RNA first. Yes or no? Yes or no? In the case of eukaryotes, you know that the RNA that forms, it is the immature RNA. What is it? It is the immature RNA. है ना Nandini, it is the immature RNA. So when we do the splicing, I told you this thing before, right? So you know that exon intron will be there. Exon intron will be there. So we need to remove the introns first, and then the heterogeneous nuclear RNA will be converted into the mature RNA. Isn't it? So at this level also we can regulate the expression. I told you, right? It's not mandatory that always we just need to remove the introns and then to join the exons. We can even join the 2 exons, the 3 exons like this. Okay, so this is the first part. Then processing level, obviously the splicing. I have covered these 2 points all together. Now, when you are transporting the mRNA from nucleus to cytoplasm, there also you can control the gene expression in eukaryotes, and finally at the translation level. But in the case of prokaryotes, it is not that much diverse. Is that clear? So genes expressed, genes in a cell, they are expressed, they will perform a particular function, right? So like see, they have given you one example that let's say if there is an enzyme beta galactosidase, beta, the name of the enzyme is beta galactosidase. Okay, so you know that this is the enzyme which can hydrolyze your lactose. If you know it, very good, otherwise note down. So this beta galactosidase, why this beta galactosidase can break this disaccharide? Wait, let me tell you. Because this lactose is made up of glucose and galactose. Okay, this is made up of glucose and galactose. So it is galactosidase. So it can break it. Okay, it can break it, isn't it? Hydrolyze it. So this enzyme, I told you, it is synthesized by E. coli. It can catalyze the hydrolysis of disaccharide lactose. So this is important, bache. Please pay attention, right? So lactose can get converted into galactose and glucose with the help of this enzyme. So bacteria can use it as a source of energy, right? So now here you know what are they trying to do? They are just trying to explain the operon concept, right? And that I am going to explain you. But before explaining the operons, I will tell you about the Human Genome Project. See, bache, the operant topic, this video, right? This individual video I have posted on our channel, right? If you will go to that video column, you will find the video on operants. Fine, you will find the video on operants. I will ask my team to pin that particular link in the description box. So firstly, I will finish Human Genome Project and the DNA fingerprinting. So if the time will be there, then I will go for the operant topic because I have already covered it. Okay. So by tomorrow, I will pin the link of that video in the description box of this video. But first, let's finish the Human Genome Project. Then I will explain the operant to you if we'll get the time. Is that okay? Yes, tell me quickly. Is that okay? Be quick. Fine. Okay, so this topic is very important. As I said, bache, from Human Genome Project, from genetic code, for sure you will get one MCQ. For sure you guys are going to get one MCQ from these topics. For sure. You cannot skip. You should not skip Human Genome Project ever. Okay, and even it is important for the CBSE board as well. Now come to this. See, many years ago, scientists, they invested a lot of money in a project. Why? Because they want to study our DNA. We know that our DNA is having the sequence. It is having the nitrogenous bases. It is having a proper sequence. We know that. But how that sequence is there on, what kind of sequence is there on DNA? We know sequence is there. We know nitrogenous bases are there. But we don't know exactly that sequence. The sequencing of that nitrogenous bases is not there. Let's say, imagine if I get to know about the DNA. If I get to know about the sequence of DNA, that let's say I am the healthy individual, there is another person who's having a disease or a disorder. So if I will compare the sequence of both the individuals, I can figure out that if that person is having a particular disorder, it is because of this and that reason. It is because of this and that mutation, isn't it? Let's say if in a healthy individual, the sequences, it's a very lame example that I'm giving you right now, just to explain it to you. Let's say it's DNA. So it's going to be A, T, C, G, C, let's say. And in a person having disorder, the same sequence is A, C, C, G, C. So if I will compare it, I can simply figure out that, okay, there is a difference of a base. है ना? So studying the human genome can help us. Studying the human genome can help us. If we'll get to know about the sequence, we can treat that disorder, that disease in a better way. We'll get to know that how many genes are there in our body. We'll get to know about their function. And even after studying all that things, we can use that sequences, right? We can synthesize some proteins in the lab conditions also. Don't you think so? Don't you think so? Yes, we can do so as well. So yes, it is very beneficial to study the human DNA, and that's what we are going to discuss in this Human Genome Project. Okay, that's what we are going to discuss here in this Human Genome Project. So in the preceding sections, we have learned that it is the sequence of base in DNA which will determine the genetic information, of course. Right? The DNA sequence will tell us about the genetic information. It will tell us about the proteins. It will make the enzymes that will be used for our metabolism. Isn't it? Isn't it? So in other words, genetic makeup of an organism, it lies in the DNA sequence. Fair enough? Fair enough, guys. I want you people to revert quickly. Okay, of course. Of course. Now, if two individuals are different, then their DNA sequence should also be different. This is what we believe. But after studying Human Genome Project, we got to know that 99.9% we all are same. Right? It is the difference of only 0.1%, and that 0.1% difference is going to, right? There is a difference of only 0.1%, and imagine because of that 0.1% we are so different from each other, right? Me, Kaustuba, Nandini, Kumar, Jay, we all are 99.9% same, right? And because after, and how do we know about all these things? Because of Human Genome Project. So it is something very important and you should know about it. Fine, you should know about it. Clear, bache? So these assumptions, they led to the quest of finding out the complete DNA sequence. So with the establishment of genetic engineering techniques, it was possible to isolate and clone any piece of DNA, and availability of the simple and fast techniques for determining DNA sequence. So they started this project, right? It was launched in the year 1990. So this is important. This is important. So by this time, we had that genetic engineering technology, right? And even the DNA sequencing ways were there. Okay, bache, DNA sequencing ways were there. So it is a mega project. You can imagine the magnitude and requirement. See, human genome, you know that in haploid individual, it is 3 into 10 raised to power 9 base pair. You know it is 3 into 10 raised to power 9 base pair. And in diploid, it is going to be, in the diploid individual, Subhashini, it is going to be, quick, in a diploid individual. See, in the case of humans for haploid, right? Right, in the case of humans, for the haploid, you know na, this diploid and haploid part. So haploid set is having 3 into 10 raised to power 9 base pair. What about the diploid set? Yes. What about the diploid set? Tell me quickly. 6 into 10 raised to power 9. Okay. So if the cost of sequencing required is $3 per base pair, imagine we have 3 into 10 raised to power 9 base pair, and for per base pair the cost is $3, right? So total estimated cost of the project was it was approximately $9 billion. Imagine, that's why it is a mega project, right? Scientists, like, see, companies, they invested a lot of money in it. Okay, so further, if the obtained sequences were to be stored in books, like if we'll get to know about these bases, about the information, we cannot store it in the books, right? We need a lot of books. And so during this process, one more field of science it emerged, and that is your bioinformatics. Okay, that is your bioinformatics. Because if we have to note down it in books, 1,000 letters, each book contains 1,000 pages, this is what you are going to get. Okay, so enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for storage and retrieval and analysis. So that's why a new area in biology developed during this period, that is bioinformatics, right? So bioinformatics is also a good career option. But if you want to go for the bioinformatics, right, make sure like you are not going to get a job directly after your graduation or something, right? You have to invest some time. And if you are really passionate about it, then you are going to get very good opportunities, right? So students, you can also search about bioinformatics. You can also search about the institutes, right, where you can do the bioinformatics. So go for it, okay? Go for it. And the competition is not too much here in this field. Fine, if you are the best here, you are going to get wonderful job here. So what are the goals of HGP? Okay, what are the goals of HGP? So the goals are identify all the, this is what they were thinking. Identify all the approximately 20,000 to 25,000 genes in human DNA. It was believed that 20,000 to 25,000 genes are there in human DNA, and to determine the sequence of that 3 billion chemical base pairs that make up human DNA. So don't make mistake here. It is 3 billion, not million. So kindly avoid such mistakes. Okay, kindly avoid such mistakes. Then store this information in databases. Finally, improve tools for analyzing that data, and then to transfer the related technologies to other sectors, right, bache, and address the ethical, legal, and social issues that may arise during this project. So these are the goals of this particular project. Clear, bache? These are the goals of this particular project. So Human Genome Project was a 13-year project coordinated by US Department of Energy and National Institute of Health, bache, right, bache? So see, during the early years of HGP, the Wellcome Trust became a major partner. Additional contribution came from France, Japan, Germany, countries like this. Clear, bache? So right, so they all contributed in that. So you can see, right, you can see the amount of money invested in this project. Imagine. Okay, so finally it was completed in 2003. So knowledge about the effects of DNA variations among an individual can lead to the evolutionary new ways to diagnose, to treat, and someday prevents the thousands of disorders as well. Clear, bache? Clear, bache? So in addition to providing clues to understanding human biology, learning about non-human organisms, DNA sequence can also lead to the can also lead to an understanding of their natural capabilities that we can use, right, in our healthcare, in our agriculture as well. So this is another point that you need to understand, that many non-human model organisms, right, the question can come from this part, bache. Question can come from this part. So many non-human model organisms like bacteria, yeast, your C. elegans, it is your Caenorhabditis elegans, right? We used to call it as C. elegans. It is actually a non-pathogenic nematode. So Drosophila, plants, etc., they have also been sequenced. So not just humans, but bacteria, yeast, C. elegans, Drosophila, plants, right? They were also sequenced. Clear, bache? They were also sequenced. So question can come from this part as well. Fine, the question can come from this part as well. Okay, so next is methodologies. These are two important methodologies that you need to remember. EST and sequence annotation. Question can come from this part. EST and sequence annotation. So firstly, let me explain this. So by this time, the genetic engineering was advanced. Okay, this is what you need to keep in your mind. So when we discuss the genetic engineering, I hope you know about the vectors, right? Like in the case of bacteria, there is one extra chromosomal DNA. We used to call it as plasmid. We have the extra chromosomal DNA. We used to call it as plasmid. What we used to call it? We used to call it as plasmid. So it is a vector, right? We can insert our gene of interest here, and this plasmid is having its own origin of replication. So it can replicate this gene of interest, it can form its multiple copies, and that can be used to make the protein, right? That can be used to make the protein. I hope you know about this. Now, why am I telling you this? Because in Human Genome Project also, they have used certain vectors. Like if I talk about the plasmid, right? It can acquire a gene having certain bases. Okay, but here in HGP, we need the DNA having more bases, large DNA sequences we need to replicate them. So in the case of Human Genome Project, they use BAC and YAC. That's a very important question, bache. That's a very important question. So BAC means your bacterial artificial chromosome. And YAC is your yeast artificial chromosome. So these two vectors, they can accommodate the DNA sequences of large molecular weight, or you can say that having large bases, having more bases. That's why they have used these two vectors. So this is a direct question. Now, I was telling you about the techniques. One is EST, that is expressed sequence tagging. Another is sequence annotation. See, you know that there are two ways. Your DNA can form the mRNA, and that mRNA can form the protein. So this mRNA is actually showing that part of DNA which is having information, yes or no? This mRNA is actually showing that part of DNA which is having the information, which is having the information to form the protein, yes or no? Of course, yes. Of course, yes. So in expressed sequence tagging, they go for these mRNAs, right? These mRNAs will be studied. These mRNAs we can study. Why? Because these mRNAs are showing us that part of DNA which is having the information for protein synthesis. But when it is the sequence annotation, the entire genome will be sequenced. In expressed sequence tagging, only these mRNAs, which is right, only these mRNAs will be sequenced. But in sequence annotation, the entire genome will be sequenced. Okay, the entire genome will be sequenced. You can see here. See, the other took the blind approach of simply sequencing the whole set of genome that contain all the coding. See, in express sequence tagging, what are they doing? One approach focused on identifying all the genes that are expressed as RNA. In EST, there are two approaches. There are two methodologies of HGP. There are two approaches. Guys, allow me a minute, please. Okay, so in express sequence tagging, one approach focused on identifying all the genes, right? On all the genes that are expressed as RNA, right? That are expressed as RNA. Like they will focus on the mRNAs, they will do the sequencing of that only. But other took the blind approach of simply sequencing the whole set of genome, the entire genome, the entire DNA, be it coding part, non-coding part, everything will be, right? Everything will be sequenced. So for sequencing, the total DNA from cell will be isolated, then it will be converted into fragments. They will take the DNA, they will cut it into fragments of smaller size, right bache, and then they will clone it. And for cloning they need the vectors, and which vectors will be there bache? Your BAC and YAC. That's important, right? That's important because they want to sequence the entire genome. Are you getting it? They want to sequence the entire genome. Are you getting it? So after it, what they get? See, so fragments were sequenced. Some Sanger sequencing techniques were used, bache, right? So the fragments were sequenced using automated DNA sequencers, right? And they work on the principle of method developed by Frederick Sanger. So he was the one who developed that sequencing method, right? It is a Sanger sequencing technique basically. So this is another important MCQ. Clear, bache? So see, the entire genome will be taken, the fragments will be formed, then that fragments will be cloned and their sequencing will be done. And which technique will be used? Sanger sequencing technique. Clear, bache? Clear, bache? So Sanger is also credited for developing method for determination of amino acids in a protein. Okay, so HGP topic is actually very important. You should not skip it, right? You should not skip it. So these sequences were then arranged, bache, on the basis of some overlapping regions present in them. Like they studied different fragments, they studied different fragments of the DNA, then they tried to arrange it, right? They tried to figure out a pattern. So as per some repeated sequences, like let's say even if you are going to sort out something, you will try to notice a pattern there, isn't it? You will try to notice a pattern there, that what kind of pattern is present there, and accordingly, right bacha, accordingly what will you do? Accordingly you will try to arrange it. Same is the case here. So after that sequencing, what they did, right, they arranged bases on some overlapping regions present in them. So this required generation of overlapping fragments for sequencing, right? So they tried to arrange it on the basis of some overlapping regions present in them. So this required generation of overlapping fragments for sequencing. So alignment of those sequence was humanly not possible, of course, bache. So they needed the programs and everything for that, and then they studied it, then they arranged it like this. Clear? Clear, bache? Right? So these sequences were subsequently annotated and were assigned to each chromosome. Like in this chromosome you have that particular sequence, in this chromosome you have that particular sequence. It is the meaning here. Okay, so this is what they've figured out, that bache, the sequence of chromosome 1 was completed only in May 2006. Imagine, they completed the project in 2003, but that sequencing was completed in May 2006. And this was the last of 24 human chromosome, right? You know, not 22 allosomes, X and Y then, okay, X and Y then. So now there are certain points that you need to remember related to this. So HGP is important. At least one question directly it will come from HGP. Directly it will come from HGP. Okay. So another challenging task was to assign the genetic and physical maps on genome. Okay, fine. They have the database. They tried to sequence the DNA and then they annotated it with chromosome number. But the another thing is that how to assign genetic and physical maps. How to do that? Clear, bache? So this was generated using information on polymorphism of restriction endonuclease recognition site, right? This, bache, this is important. Please pay attention. See, another challenging task. That's why, you know, I took the snips of the NCERT so that I can explain it. So that's important. Please pay attention. Another challenging task in Human Genome Project was assigning the genetic and physical maps on genome to that haploid content. They were just trying to assign the proper mapping so that they can even check the distance also, right? Things like that. So this was generated. How they accomplished it? This was generated using information on polymorphism of restriction endonuclease recognition site and some repeated DNA sequences known as microsatellites, right, bache? Microsatellites. So imagine. So during this process, this is what they observed. You know restriction endonucleases, This is a specific enzyme which will recognize a specific sequence and then make a cut in DNA. We will discuss that in biotechnology. I hope you know about the EcoR1. Right, I hope you know about the EcoR1. Clear, bache? So that enzymes are going to make the cut and all. Clear, in a proper pattern. It will, that restriction endonuclease break that sugar phosphate backbone. So for assigning the genetic and physical mapping, what they did, they used the information on polymorphism of that recognition sites which are present in our DNA, and our DNA is having that sites, and some repetitive DNA sequences. So yes, these all were the findings of this HGP, that our DNA is having some repeated sequences, right? That repeated sequences, they can vary in different individual. Like maybe there is a like in my case, let's say there is a sequence. In your case also the sequence will be same, but it's just that only 1 or 2 bases might be they are changed like this. And that changes we have to notice. So we discuss it. So that repetitive DNA sequences are nothing. These are the microsatellites, these repeated DNA sequences. So this is important. So one of the application of polymorphism in repetitive DNA sequence, we will explain it in DNA fingerprinting. Right? So these findings, see, during this project, bioinformatics came. During this project, they got to know that we are 99% same, 0.1% different. So we got to know about DNA fingerprinting as well, which is very important, right? Which is very important. So yes, overall, it project helped a lot to understand all that, arrangement, and even it helped in studying the various disorders as well. Okay. So these are the salient features of the HGP that you need to remember. Okay, these are what the salient features of the HGP that you need to remember. So bache, HGP, as per this, human genome contains 3164.7 million nucleotide bases, right? They were expecting something, and and these are actually the results. Okay, so 3164.7 million nucleotide bases. So in the final NEET paper, instead of million, they can mention billion. So do not get confused. It is 3164.7 million nucleotide bases. Important it is. So average gene is having 3000 bases, but sizes vary greatly, with the largest known human gene, which is dystrophin, at 2.4 million bases. So on an average, they were saying the gene is having 3000 bases, but the largest human genome. The another question, which one is the largest human gene? It is the dystrophin. It is the dystrophin. It is related with that muscular dystrophy, if you can link. Yes, it is related with that muscular dystrophy, if you can relate. So the dystrophin is having 2.4 million bases. So each and every point here is important. And total number of gene is estimated at 30,000, much lower than previous estimates of 80,000 to 1,40,000. So this is what they got, right? That near about 30,000 genes are there, bache, and almost 99.9% nucleotide bases are exactly same in all the people. So it is a difference of only 0.1%. Okay, so functions of over 50% of the discovered genes, they are still unknown, right? And in our case, bache, only less than 2% of DNA is giving the information to form the protein. Imagine. From this HGP, we got to know that we are still not aware about the function of above 50% genes. We don't know about the function of that 50% genes. Okay, and moreover, after this HGP, we got to know that less than 2% of the genome codes for the protein. So whatever enzymes are there in our system, whatever proteins are there in our system, so less than 2% of genome is coding it. And even in the DNA, that repetitive DNA sequences are there, right? That sequences are there, that polymorphism is there. So see, repeated sequences make up large portion of human genome. And these repetitive sequences, they are nothing, but they are the stretches of DNA sequence that are repeated many times, sometimes 100 to 1000 times, and they are thought to have no direct coding function, but they shed light on chromosome structure, dynamics, and evolution. So in our DNA, we have the repetitive DNA sequences, which will tell us about the evolution. They are not going to provide us any information for the protein coding. Clear, bache? So chromosome 1 is having most genes, 2968, and Y has the fewest. Important. So scientists, they have identified about 1.4 million locations. Like this is our DNA. So they figured out 1.4 million location where they have seen the polymorphism. Okay, see, where single base DNA difference, like let's say we are comparing our DNA, right, Nandini? We are comparing our DNA. So when we are comparing our DNA, 99.9% it's same. But there are certain points where we will see the polymorphism of a single base. Like might be in your case at that place A is there, in my case G is there. Like this, something like this. So what we are calling it, see, this point is very, very, very important. Scientists, they have identified about 1.4 million locations where single base DNA sequences occur in human, and this information promises to revolutionize the process of finding chromosomal location for disease associated sequence and tracing human history. So it is helpful in that way. And that single nucleotide, right, because in our cases in that DNA there are different positions where a single nucleotide is getting changed, so that is known as SNP, single nucleotide polymorphism, and which it is pronounced as SNPs. Okay, SNPs, they are pronounced as SNPs. Now I'll relate it. Yesterday I was telling you about the spliceosomes, right? I was telling you about the spliceosomes. So there we were talking about the small nuclear ribonucleoproteins. So they were pronounced as SNRPs, SNURPs, right? These SNRNPs, they were pronounced as SNRPs, but this single nucleotide polymorphism, it is pronounced as SNPs. So do not get confused here in the SNRPs and the SNPs as well. Okay, so SNRP is different, SNP is different. SNP is the single nucleotide polymorphism. SNRP is, you are talking about small nuclear ribonucleoprotein. Okay, you are talking about small nuclear ribonucleoprotein. Is that clear? Is that clear? Sure. So application and future challenges. So see, deriving meaningful knowledge from DNA sequence will define the research through the coming decades, leading to our understanding of the biological system. So imagine in our system, we are not aware of the function of the genes, right? We don't know the function of all the genes in our system. Imagine. Okay, imagine. So this enormous task will require the expertise and creativity of tens or thousands of scientists from varied disciplines, right? So basically we all need to join in this mega project, and then further, we can decode more and more information. Clear, bache? More and more information, that's what they are trying to say. Okay, and because of this we got to know about what? About the DNA fingerprinting as well, right? It's a very, very, very simple process, the DNA fingerprinting. So it was invented by Ellis Jeffrey. Okay, this technique was invented by Ellis Jeffrey. And if you talk about in India, right, it was started by Dr. V.K. Kashyap and Dr. Lal Singh. In India, it was started by, okay, Dr. V.K. Kashyap and Dr. Lal Singh. So, bache, that SNRPs that we were discussing now, so they are somewhere helpful in this DNA fingerprinting. Are you getting it? That SNRPs are helpful here in DNA fingerprinting. So, see, 99% of the base sequence in the human, it's same, right? It is same. The difference is of only 0.1%. So it is these difference in sequence of DNA which make every individual unique in our own ways, isn't it? So if one aims to find out genetic difference between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Of course, we cannot just sequence it always, right? We cannot sequence the entire DNA and then we can figure out the difference. No, let's focus on that 1.1%. That will be easy then, right? So imagine trying to compare two sets of 3 into 10 raised to power 9 base pairs, right? DNA fingerprinting is very quick to compare DNA sequences of any two individuals, because here we are going to focus on that 0.1%. Are you getting it? We are going to focus on that 0.1%. Is that clear? Is that clear? Tell me, is that clear? So it is because of the repeating base patterns in DNA. When it is the DNA fingerprinting, it is because of that repeated right? Yeah, repetitive DNA present in our repetitive DNA. Clear, bache? It is because of that. Okay, and here in this DNA fingerprinting, we also talk about VNTRs. We also talk about RFLPs. Do you know about it? Yes, bache? Actually, in DNA fingerprinting, you are going to talk about two things, VNTRs and RFLPs. VNTRs means variable number tandem repeats, right? These are kind of mini satellites. They were discovered by Alec Jeffrey. And RFLP is your restricted fragment length polymorphism. Restricted fragment length polymorphism, right? What is this RFLP? It is your restricted fragment length polymorphism. So let me explain this part to you. Okay, okay, yes bache? See, it's very simple. I told you VNTRs were discovered by the Alec Jeffrey, and these were the kind of mini satellites. So satellite DNA, I hope you know, it is the result of polymorphism. Here we are talking about that repetitive DNA. It's very simple. You know that we have exons as well, we have introns as well, isn't it? I hope you know that we have the exons as well, we have the introns as well. So bache, exons are the coding region, right? They have the information to form the protein, and introns, they are what? They are the non-coding regions, isn't it? What are they? They are the non-coding regions. They do not have the information. They do not have the information, right, for synthesizing the protein. But in the case of introns, you will see some repetitive DNA sequences, right? You are going to see some repetitive DNA sequences. Let's say 11 to 20 base long, right? Let's say 11 to 20 bases, they are repeated so many times. They are repeated so many times. Yeah, 11 to 60 base pairs. Okay, okay. So in general, when we see this, right, so these VNTRs, they are nothing, they are the mini satellites. So they are basically having the repetitive DNA sequences. You can say 11 to 60 base pairs, right? 11 to 60 base pairs, which are repeated again and again. It is written a variable number tandem repeats. So these repeats, this repetitive sequence will be repeated again and again. Clear, bache? Clear, bache? Done? Done, done, done. So basic repeat sequence of 11 to 60 base pair is there. Okay. So this repeats, they show a degree of polymorphism. Do you know that? These repeats, right? These repeats, they show a degree of polymorphism. It's not like that these repeats are same in your case and in my case, right? If we belong to the same family, so might be there can be some similarity, but still it is not totally same. In the case of your monozygotic twins, right? In the case of monozygotic twins also, we can still consider that similarity is more, but it is not actually like that, right? So these repeats, their size, it varies from individual to individual as well. Do you know that? These repeats, their size, even it varies, right? It varies in individual to individual. Clear, bache? Clear, bache? Right? So the number of these repeats, because these are the repetitive DNA sequences. What are they, Sabha? They are the repetitive DNA sequences, right? And here, I told you before that we have the, even in NCERT, it was written a restriction endonuclease enzyme. So, bache, restriction endonuclease enzyme is the one which recognize these repeated sequences. Even you will talk about the palindromic sequences as well when we will discuss it. So, now, as I said, repetitive DNA is there. It is having 11 to 60 base pair, which will keep on repeating, right? So obviously that repetitive DNA is having the sites where you can, use your restriction endonuclease, where you can put the cuts, okay, where you can cut it. So obviously, if you will compare it in different individuals, the size of that fragments and number of base pair, it is different, right, Sabha? Right? In your case, that repetitive DNA, the size of that repetitive DNA, the sequence of that repetitive DNA will not be same, right? Will not be same. It is not going to match with another individual. That is your uniqueness. That is your uniqueness, and that thing is basically restricted fragment length polymorphism, right? We discussed it there. Clear, bache? So when genome of two people are cut using the same restriction enzyme, the length and the number of fragments obtained, they are different. Okay, they are different for both. So when we cut the DNA of two different individuals with same restriction enzyme, the fragments you are going to get, their size and sequence, it is different. That comes under this restricted fragment length polymorphism, and these two things are used in this DNA fingerprinting. So ultimately DNA fingerprinting is based on this polymorphism which is there in our DNA sequences. It is based on this difference which is there in our DNA sequence. Is that clear? Yes, bacha, is that clear? Sure? Sure? Okay. So see this. So DNA fingerprinting involves identifying differences in some specific region. You know that it is the repetitive DNA, because in these sequences a small stretch of DNA is repeated many times. So this repetitive DNA are separated from bulk genome DNA. So what are we going to do in DNA fingerprinting? That repetitive DNA sequences will be separated from that bulk DNA, right, bache? And during density gradient centrifugation. How? During density gradient centrifugation, Sanjan. Okay, so bulk DNA, the major DNA will form major peak, and other small peaks are your satellite DNA. So because of that repetitive DNA, the peaks that you are going to get due to this density gradient centrifugation, it is your satellite DNA. It is your satellite DNA. So depending on the base composition, bache, AT rich or GC rich, length of segment, number of repetitive units, the satellite DNA, it is classified into different categories like microsatellite and mini satellite. Like in the case of mini satellite, I told you, 11 to 60 base pairs, they will be repeated. So this microsatellite is your homework. You will tell me what is it. Okay. Okay. So on the basis of number of bases and the length of that fragment, we are dividing it like this. Right? So these sequences, they are not going to give any information for protein synthesis, bache, but this high degree, they show high degree of polymorphism, and it is the basis of DNA fingerprinting. Is that clear? Is that clear? Right? So since DNA from every tissue, right, blood, hair follicle, but not from RBCs. RBCs, if they are immature, they have nucleus, then they have DNA, otherwise they do not have. Clear, bache? So from the individual, it will show same degree of polymorphism. They become very useful identification tool in forensic application. So I hope you know that. And these polymorphisms, it is also inheritable. So that's why in the family history, you will see more similarity. But still, it is not 100% same. It is not 100% same. Is that clear? So as polymorphism in DNA sequence is the basis of genetic mapping of human genome, as well as of DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. Now, see, this portion, the question was there in the paper and we all got confused. I think it was in NEET 2022, right? Right. So polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting. Yeah, still remember. So even many teachers, even I made the mistake here. I just marked that polymorphism is the basis of DNA fingerprinting, but this polymorphism is also the basis of genetic mapping as well. Okay, it is also the basis of genetic mapping as well. So it is MCQ. I think it was in NEET 2022. Do check it out. Fine. Do check it out and solve it. Done, bache? Done, bache? Clear? So, new mutations may arise in individual, in somatic cell or in the germ cell. You know that. So, if germ cell mutations are there, right? So, obviously, it will be transferred to the next individual. So, that's it. That's all. So, here, this is the part majorly that you need to know. Clear? Clear, 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 clear? Okay. So, see, alleles, you know na, allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant at a locus occur in human population with a frequency greater than 0.1%. Here they are just trying to explain that polymorphism. You talk about the DNA, you talk about the genes, right? And in the genes, we talk about the alleles. In the genes, we talk about the alleles. So alleles are what? when there is a sequence change, when there are certain changes in the gene, it can result in the form of its variant, that is allele. And you know that we have multiple alleles as well, like blood group. In human population, you see more than 2 alleles for that. So here, this is what they are saying, that allelic sequence variation has traditionally been described as DNA polymorphism, right? If more than one variant at a locus occurs in human population with a frequency greater than 0.1%, 0.01%. So if frequency of that allele in human population, like at one locus, if it is more than one, so this is what they are showing, that it is also the DNA polymorphism. Clear? So in simple terms, if an inheritable mutation is observed in a population at high frequency, it is DNA polymorphism. Right? If any inheritable mutation, like if in our DNA, Bache Kaushik, they are trying to relate the polymorphism with the mutation. Right? They are showing that polymorphism is the base for fingerprinting, and how that polymorphism is there, it is actually the mutation. And what type of mutation? You know, mutations are in somatic cell also, in germ cell also. So if mutations are in germ cells, that is, if they are in our gametes, obviously they will pass to the next generation. And during that mutations, what is happening? Right? The sequence is ultimately changing. Right? The sequence is ultimately changing, and obviously it is also forming the alleles. If there is any change in that gene sequence, it is basically doing what? Alleles. It is forming its variant. It is forming allele. That's what they are trying to explain here. And you know that type of mutations, they are inheritable and they are responsible for polymorphism. Like if in my DNA there is some mutation in my gametes, obviously it will pass to my next generation. Clear? So probability of such variation to be observed in non-coding DNA would be higher, right? Right? So these mutations kept on accumulating generation after generation. Such mutations, they will keep on accumulating, right? And they are responsible for this polymorphism. If we are saying now the polymorphism is there in our DNA, why? Because of mutations. And once they will get accumulated. Clear, bache? Clear, bache? So single nucleotide polymorphism can be there, and further other things are also there here. Okay? Okay. So now when it comes to the technique of the DNA fingerprinting, so you know the scientist's name, bache? So what he used, I told you already, variable number tandem repeats. And variable tandem repeats, they are nothing, they are the mini satellites. Right? They are the mini satellites. Clear? Clear. So here they have even used the technique of Southern blotting. Right? And even if you see, first of all, you need the DNA for this technique. You have to isolate the DNA. If you are getting the good amount of DNA, then it's fine. But if not, you can use polymerase chain reaction to form the multiple copies of that DNA, isn't it? To form the multiple copies of that DNA. So isolation of DNA, then comes the digestion of this by restriction endonuclease. Restriction endonuclease will cut that polymorphic forms. It will make cut on that repeated DNAs, of course. Then separation of DNA fragments by electrophoresis. Now you have different fragments. So on the basis of size, it is not on the basis of charge, on the basis of size, with the help of this electrophoresis, you will separate it. In electrophoresis, you know your DNA is negatively charged. Why? Because of phosphate, right? So electrophoresis is a technique in which electric field is applied and the charged molecules, they will get separated. How? On the basis of size, right? So the one, because DNA is negatively charged, so it will move towards, right? It will move towards anode or cathode. That's your homework, check it. DNA is negatively charged, where will it go? It will move towards anode or cathode. That's your homework, do check it. Clear? So finally separating that DNA fragments, they will transfer, right? The transferring or blotting of separated DNA fragment on synthetic membrane. Like they will, let's say here you have the fragments, right? And then you are taking it on another, right? Another membrane, synthetic membrane, like your nitrocellulose or the nylon, right? You are going to take it like this, right? Like we trace something, no? Same way. So then hybridization will be done by using VNTRs, variable number tandem repeats, because we already know about it. So we will hybridize it with VNTRs, and accordingly, bache, then it will be detected. So as of now, I'm not explaining it in too detail. The reason being is you really need to know about the other techniques also. Like you should know electrophoresis in detail. You should know about the PCR as well. You should know about the Southern blotting as well. So we will cover that part in biotechnology. Okay, so once we will finish the biotechnology, you will understand it in a better way. Okay, then again I will elaborate this topic to you there. Okay, so blood sample, DNA is extracted. So see this, we will make the fragments of it, and then we are going to separate that fragments in electrophoresis. Then like we used to trace the paper, no? So same way we will use that fragments, we will put it on nitrocellulose or nylon sheet. This is Southern blotting. Southern blotting is used for DNA. Northern blotting is used for RNA. It's like that. We will discuss it in biotech. So see, the DNA band pattern of the gel is transferred to nylon membrane. It is Southern blotting. And here to these bands, VNTRs will be added. They are acting like a probe, right? Right. So DNA probes, these VNTRs will bind to that repeated sequences, and then bache with the autoradiography, you will check that bands. Okay. So there we will discuss it. Okay, there we will discuss it. Is that clear? So that's all about this chapter, bache. The topic of operance. Okay, even the topic of operance, I have posted one video on it. So that's my advice. You should check that video. Okay, you should check that video. And today, because we are done with the chapter, so I'll share all the PDF files in the Telegram group. Fine, I will share all the PDF files in the Telegram group. So one more chapter done, genetics, principles of inheritance and variation done, molecular basis of inheritance done. Next, do you want to start evolution or you want to start some another chapter? Tell me. Right, in the comment section, do let me know which chapter you want me to start. Accordingly, I will share the schedule with you all. Right, tomorrow there is no class on YouTube. Okay, and you have to utilize this time to check that operand video because that's important. Right, that's important. So do revise this part, practice the question. If there is any doubt, just put that doubt in the comment section. Okay, I will answer that. For sure, I will answer that. Okay, okay. So just give your best. You still have 6 months, bache. Yesterday I have taken a session live to explain the differences in your old syllabus and new syllabus, which book you should follow, right, which topic you should do first, which topics you should avoid. So do check out that video. And yes, now you know what you have to do. In the comment section, do let me know that you like the session or not, right? Just let me know about your views, and do let me know which chapter you want me to start next. Okay, okay. So take care, everyone. Thank you so much for watching. Stay tuned, and if you want to join our batch, the link is there in the description box. Thank you.